Okay, so good morning, everyone, and I'm very pleased to the
population. So is Qatar suitable for these kinds of studies? And this is where I would say that yes, even though the population is small, as a total population, there's a lot of geographical and cultural isolation over the years, which caused a lot of inbreeding, as I've showed you. We have very large families, so we get a lot of recessive alleles. We also see more children. With more children, you always get the risk of de novo disease. Because we have clustering in families, we end up getting these Mendelianized forms of complex disorders. And because of the high rates of consanguinity, which is up to 50% of marriages are between first and second cousins, we're very well suited to discover new genes and recessive alleles and also to look at human knockouts. So that's where NGS comes into play. These are just some examples of some of the pedigrees that we work with. At Sidra, we've developed this this pipeline where patients walk into really high quality clinical care. They're enrolled into research, and then once they're enrolled into research, they're consented to do pretty much anything to their samples in order to discover what's wrong. And then we try to bring this back to the clinic to lead to precision medicine or personalized medicine, if you will. So we've now initiated this across multiple clinical departments, where we identify subjects. We refer them first to pathology to rule out anything known, for example, gene panels or exomes. And then if they're still idiopathic, we recruit them for research. And of course, in research, we try to collect as much sample as possible, and we store it long-term in a repository. And these repositories will be open to collaborators if anyone wants to get involved in studying a particular disease. We then start from whole genome sequencing. As I said before, at Sidra, we run the National Genome Sequencing Reference Lab. So we're very well equipped to do this at scale. We've developed our own bioinformatics programs, not only to look at small variants, for which best practices exist, but also to look at that we think is still a real de novo mutation. And of course, the more siblings you add, the larger that effect. Similarly, for homozygous mutations, this is where we have a recessive family with multiple affecteds. We have hundreds of mutations per child, but the more children who have the disease, if you look at sharing, then you only get a fraction of those that are shared by another child. And then again, shared by 3 siblings or shared by 4 siblings. So in this case, for example, you would take hundreds of mutations and you would only have 1 that's left. And it's usually the causative one because it's shared by these 4 siblings and it fits. So this is the importance of sequencing everyone in the family that you can enroll, and of course having good population match controls. So let me show you examples of some diseases that we've solved this way. So this is a kid who had the congenital diarrhea syndrome. They were born preterm. They've had multiple lost pregnancies, and of course, unfortunately for the family, 3 successive newborns that died because of this condition. Using next-gen sequencing in the NICU for this child, we discovered this stop mutation in MYO5B, which is known to cause congenital diarrhea syndrome. Similarly, here we have congenital surfactant deficiencies. These are kids that are born with an inability to breathe properly because they lack surfactant. We end up finding a novel Qatari-specific mutation in this child. Again, these are 3 siblings that develop Batten disease, which occurs fairly late in adolescence, and really it's a debilitating neurological condition. And so again, by sequencing, we find these mutations that segregate. Again, when we have multiple affecteds, you can find these mutations very, very effectively. What we also discover is something like this. This child came in before. This is another child that came in with the same exact phenotype, but from a different family. When we sequence them, we actually find the same exact rare mutation. And this is actually not uncommon. We look at here also. Here's 2 families that came in with Alport syndrome, which is this nephrotic condition. Again, they came in separately. And they both ended up having the same exact frameshift mutation in collagen 4A. So structural variants, for those of you who might not know, are basically deletions or insertions in the genome that end up causing chunks of the genome to be absent or to be in multiple copies. There's also more complex structural variations like chunks of the genome flipping or inserting themselves or translocating to a different chromosome. And so we may know clinically of many disorders that are caused by structural variants. For example, Down syndrome is a duplication of an entire chromosome, but there are smaller events also. And we know that about 10%, 5 to 10% of our genome is structurally variable, and much of this is just polymorphism in the background, like SNPs, but some of it could be pathogenic. So we've actually developed, through our whole genome sequencing pipeline, a homegrown method to find structural variants. And so we're going to publish this soon. But basically, here's some of the variants that we find. So this is a child with autistic behavior and developmental delay without any family history. When we sequenced the parents and the family, we found no point mutation. We then ran our structural variant pipeline. We found this deletion that occurs on chromosome X. So as you can see, the mother is a carrier of this deletion, so she's asymptomatic, but the male child is hemizygous, so he has this deletion here. And so this deletion actually removes exon 1 of the POF1B gene, which is already known to cause autism. This is another example, actually, a very interesting example, because this is a family that has cleft lip and palate, skin and genital abnormalities, and short stature. And everything was tried with this family. So they've been diagnosed with this syndrome, but they've been negative for all the genes of this syndrome. Microarray was negative, whole exome sequencing was negative, whole genome sequencing was negative. We didn't know what was wrong with this patient. And through our structural variant pipeline, we actually discovered this event. So as you can appreciate here, there's no deletion or duplication, but there appears to be some form of rearrangement. And so we used a de novo short read assembly approach to actually reassemble the short reads around this area. So this is around a 500 kb area. And what we discovered was actually really, really interesting, because it turns out that this is a reference allele in this order from left to right. And what we have is actually a deletion that occurs upstream that's then inserted downstream. And so this deletion...
Well, you've been so deep in this Qatar genomic program. And if you come back like 6 years back, would you change something? Because you observe how this genomic program in Qatar developed. So would you change something in the beginning to get better result now? So if you will be starting like at the moment, what would be the difference from what we have today? Oops, it looks like we have a connection issue.
Can you hear me?
Yeah. So
your question was, would I change something, right?
Yes. Oh, it's all goes smooth and very nice so far. So how do you see it?
Yeah. So I would say, probably I would say that perfect is the enemy of good enough. And I would say that, instead of just planning and trying to plan how we're going to do it, just start somewhere, and I would really encourage starting grassroots, like maybe on a very small level in certain universities, people can start doing some sequencing projects. But the most important thing I would think is get the consent right, because the patient consent is basically what, you don't want to be in a situation where you've sequenced 6,000 to 10,000, and then you realize that we can't go back and ask something. It's very, very debilitating. So the way we did it was we just started by sequencing, and then over the years we developed this Mendelian program, and we amended our protocol to actually start biobanking more samples, getting access to clinical data. Of course, all of this is anonymized and it's protected clinical information, but just getting started is the most important thing.
Clinical variables for diagnosis of lymph node metastasis and endometrial cancer. And the uniqueness of this talk, but it comes from pure medical needs. So Yoshida-san is a doctor who just initiated very interesting and contributing research. And Yoshida-san, please, you have maybe 25 minutes, and please leave a few minutes for answering your questions. Thank you. And don't forget, unmute your phone. Good. Yes.
Thank you, Oleg-san. Can you see my slide? Okay.
Very nicely. Very nicely. Okay. Yes.
Thank you. Let's start my presentation. It is a great honor to be given this great opportunity to present here. Thank you very much. I'm Emiko Yoshida from Juntendo University in Japan. Today, I will talk about our research project, messenger RNA biomarker identification and novel predictive model for endometrial cancer. Today, so my specialty is OBGY doctor. So I will present about the necessity of biomarkers from the view of a clinician. So first, I will introduce uterine endometrial cancer. The number of gynecological cancer patients in Japan has experienced in increasing trend in recent years. And here I will talk about uterine endometrial cancer. Uterine endometrial cancer, So uterine endometrial cancer develops from the lining endometrial layer. The primary treatment for uterine cancer is surgery. So uterus and fallopian tubes and ovaries and removed. Lymph nodes which have risks of cancer metastasis is removed, not only from pelvic area, but para-aortic area like this. In many cases, widespread lymphadenectomy entails patient's burden, and it is a cause of reduced post-operative quality of life. Actually, the incidence of lymphedema like this due to lymphadenectomy is over 36%. The physicians prefer to avoid irrelevant lymphadenectomy, so the percentage of operation with lymphadenectomy seems to be smaller like this. However, currently, these are no clear criteria for determining whether lymphadenectomy should be performed or not. Approximately 80% of uterine endometrial cancer are low intermediate risk group, and they have good prognosis. In addition, in this group, lymph node metastasis complication is at approximately 10%, and lymphadenectomy has no established evidence about efficacy as treatment. So there are many opinions that lymphadenectomy should not be performed on low intermediate risk patients with no lymph node metastasis. Now, list one sample. We performed a differential analysis between lymph node metastasis positive and negative groups, and we found 163 promoters as differentially expressed. Then we narrowed down the candidate with detectable expression levels and substantial difference between the groups. In other words, median expression was more than 16 CPM at least. Fold change are more than 16, and AUC was more than 95%. AUC means accuracy index. And we identified 6 candidates remained like this. And then we validate candidates genes by QRT-PCR in 115 clinical samples. As a result, we identified the most significant candidates genes. One is SEMAS3D, and another was novel isoform THCC2. The red mark shows the lymph node metastasis positive samples. In this positive group, SEMAS3D expression level was lower and novel THCC2 expression level was higher. The discrimination power of combination marker of SEMAS3D and novel THCC2 is very effective like this. However, here we focused on THCC2 isoform, which was identified as a biomarker. A variety of isoforms had been reported as THCC2 messenger RNAs like this. And many promoters of THCC2 also had been identified in the Phantom 5 project. we found P10 THC2 promoter as differentially expressed. The lower panel shows the entire region of THC2 locus, and the upper panel is zoomed view of P10 THC2. Sky blue highlighted background indicates the P10 THC2 location was how to assess the risk and select diagnostic targets preoperatively. In other words, risk classification is based on the final pathological diagnosis. This final pathological diagnosis includes the assessment of histological malignancy and cancer progression. However, before operation, we can't assess about the final pathological diagnosis. So we try to predict histological malignancy and cancer progression by using biopsy data and diagnostic imaging data, but its concordance rate is not completely. And then how to predict lymph node metastasis only by using parameters which can be assessed before surgery or during surgery. We planned two methods. One is combining our biomarkers with other biomarkers, and others was to combine our biomarkers with clinical parameters which can be assessed before surgery. The cage data is a comprehensive data of gene expression profile of endometrial cancer. We reanalyzed previous cage data and compared the gene expression profiles between low, intermediate, and high-risk group. As a result, we succeeded two new companion markers. The gene name cannot be published due to the AUC of top 8 parameters are assessed almost the same like this. However, too many variables make calculation complicated in clinical use. In the view of this disadvantage, top 4 parameters may be the best combination, but at the first trial, we selected 8 all parameters. Then how to predict lymph node metastasis? Optimized models is required to evaluate the presence or absence of lymph node metastasis by using many parameters. We chose a generalized linear model because our parameters include not only continuous variables, but also categorical data. Continuous variables take consecutive values such as quantified value of biomarker, weight, or age. Categorical variables are numerical values used only for distinguishing. It only makes sense whether it is equal or not, and it does not mean how large or how many times it is. For example, pathological types of endometrial cancer. First, we try to predict lymph node metastasis using the logistic regression model, which is one of the generalized linear model. Logistic regression analysis does not directly predict whether an event will occur, but predict the probability of that event occurring. We calculated the probability by using clinical parameters and gene expression levels of biomarker. First, we try to validate the efficacy of logistic regression analysis in training data set. We had to select only low intermediate risk patients. As a second, there is a risk of metastasis until the primary lesion is removed from the patient's body. If the primary tumor tissues are collected from the removed uterus, and and the gene expression are evaluated, metastasis risk due to the difference in time will be overcome. So we suggested novel interoperative rapid diagnosis method using biomarker. Now we try to develop rapid quantification method for messenger RNA biomarkers and calculate method for predict values of metastasis. And this is conclusion. It is difficult to classify various, it is difficult to classify various clinical phenotypes by single gene expression. One of the solutions is combination with other parameters, for example, clinical parameters. Integrated analysis has a probability to establish novel diagnosis method for clinical use. And this is acknowledgement. These are our research group members. This research project is joint research with other multiple facilities like this. Thank you for your attention. Thank you very much.
Yoshida-san, thank you. I'm so happy to see the steady progress you do. So it's amazing. So we have some issue with YouTube.
This method, and try to develop the all packaging of diagnosis, and so we have to get the support by company for clinical use. So that is our next step, and the gene quantifying method is already completed, developed, and we have to improve the method for accurate diagnosis, because this method is for patients. So we have to keep the high quality and high accuracy. So we have to improve our method. So this is our purpose and our next stage. Yeah.
Thank you, Yoshida. And last question. So as I see, you return to original cage data, or you made even additional whole genome cage profiling data. So it looks like if we're starting a new project like we do in Russia, for example, it's very smart not to try PCR check by this kind of known biomarker, but it's better to have like whole genome profiling of a certain number of samples just to have a chance to get new biomarkers or make kind of wider potential for analysis. Is it true? Because it looks like having the whole genome profiling for you was very, very useful, because you got new companion biomarkers by different angle of analysis.
Yeah, so the case data is very essential for any other research project, but this it is very important to set up the two groups to compare. If we missed create the two groups and to create the gene expression levels, so the data is not enough accuracy. So we always feel very nervous to set up the groups
me do it in official way, who is our Deputy Director of RIKEN Center for Integrative Medical Sciences in Japan, and, which is very impressive for me, Head of Genomic Research of newly established quite fancy Human Technopole Center in Italy. And the talk will be on the quite impressive and quite exciting for us topic about identification of non-coding genome elements and a single cell resolution. This is era coming now for single cell genetics and genomics, and it will be talking about identification of elements in a single cell level for fine modulation of gene activity. Yes, please, Piero, go ahead. Thank you very much.
Hi, Oleg. Thank you very much. Can you hear me?
Yes, very nicely.
Okay, very good. So it's a big pleasure to be there. It'd be much better to be there in person because I never been really to Russia. But anyway, it's a pleasure to be here online for today. I'm going to talk about a part of the activity of my group and a little bit of our center. And first of all, I would like to start with a disclosure of conflict of interest, because a part of what you will hear today is a part of the activity is a part of the activity of this company that I funded, and I would like to mention a little bit about this. So the idea behind our project has been always to make a very large encyclopedias of genomic element and try to understand what happens in the genome. And probably you may have heard about those large projects like ENCODE project and the Phantom. Particularly, we have been organizing the Phantom to understand, to map all the regulatory elements, having very large catalog of cells. We have been analyzing thousands of cells, many primary cells, to really map all the promoters, enhancers, regulatory element in all of those cells, also using tissue and cell lines as well. And you can see this is a large collaboration, and you can see that we have also have a good collaboration with many of you guys, and it's very nice to really sense transcription. And this is one specific antisense, and antisense is depicted here in red, is the antisense of the UCHL1. UCHL1 is here in green, is a protein coding genes. And this is important because it's one of the Parkin, is one gene that, if mutated, causes familiar Parkinson's disease. My collaborator, Stefano Gustincic at SISA in Italy, has been studying this, and especially has been overexpressing this antisense in cells that do not express this antisense to see what happens to the sense. The sense here is in green, the protein coding genes. And we look at the messenger RNA level, and nothing happens. You can see it's actually not statistically significant. But if you look at the protein level, we find that there is much more protein produced in the presence of the antisense. So to summarize, no changes at the RNA level and dramatic increase at protein level for this antisense. And we have been analyzing a little bit more in details, and we have been noticing that there is this part in green here is a sign element shown here. If you take away the sign element, or if we flip, you put in the opposite orientation, we see that this effect to induce protein expression goes away. So essentially, we don't see this anymore. So this is a very important feature. We have additional data that shows that the antisense part is important, but I don't show all the details here for time. We've been further looking at the transcription, asking, is this a specific, a very exceptional RNA, or are there more of those RNA? And this is the work, unpublished work of Harshita. She has been looking at a series of them, and this is at least this is a short list of those that have that seem to work. So essentially, there are antisense containing a sign element, and that overlap genes, and they do work. At least half of the antisense containing a sign element induce protein expression. And this is in the mouse. But we also identify the human counterpart, overlap a co-localization in the cytoplasm, sense-antisense, using the EGFP as a target, and the scramble or delete or deletion of some of the sign elements show lack of localization. Essentially, she has been quantifying with a high resolution microscopy, showing that there is actually strong co-localization in the cytoplasm. She has been looking next to the proteome, the proteins that bind this. So essentially, she has been using this our antisense. This is our synap, and we take antisense oligonucleotide to pull out from the cells, and then with a mass spec, we analyze the RNA binding protein, and we identify a series of RNA binding protein as in particular for reliability and specificity. We have analyzed those here, and particularly PTBP1 and HRNPK were quite important RNA binding protein, quite essential for the function. And she wanted to knocking them down to see if we find decreased colocalization in absence of protein. So colocalization means that sense antisense colocalize in the cytoplasm and help, and the antisense help translation. Knocking down those RNA protein, the colocalization goes down. So essentially, everything falls off. And actually, here is also the retention in absence of those protein. The synapse just remains protein. You can see it here. In the mouse model, there is more protein, but one gene only causes less protein. And with the synapse, even if there is only one gene active, we can target and have twice the amount of protein. So essentially, rescue using a therapeutic synapse. Does it really work? And in collaboration with the Telethon Institute in Naples, our collaborators have been working to enhance the translation of COX-7B. COX-7B is important for one human diseases, is microphthalmia with skin lesions, and they develop a Medaka fish model where essentially they put a morpholino and inhibit splicing, causing exon skipping. And this exon skip is about 70% of the RNA. So there is only 30% of normally spliced protein, so 30% of protein. So if we design, and you can see the phenotype, once we design the antisense with the binding domain and our sign element here, we actually they rescue the phenotype. So the fish is again normal by adding this antisense in the presence of the morpholino. This is just adding total RNA, polyadenylated synapse RNA in the embryo of a fish. It's quite impressive. Of course, the fish is not a human, and actually we want to go on the animal model. We also been looking at different diseases that are not haploinsufficient, but diseases where we may provide a therapy by enhancing the amount of protein. One is that one thing is about Parkinson's disease, where there is a degeneration of the dopaminergic neurons, and there is actually one gene that is a GDNF that promotes survival of dopaminergic neurons. And essentially, in the mouse model, can we add synapse GDNF to the messenger RNA? And the promote is in our lab, and there is a lot of intellectual property, but I'm gonna probably skip about this. But just what we are thinking is that there will be many diseases, and from the liver, blood, brain, skin, and various other diseases that can be addressed by increasing the amount of protein. What is always very important is, however, to look at the right isoform of RNA. So what is very important is to target the antisense should target usually the 5' end of the genes. Here is a typical one target is hepatocyte nuclear factor 4 alpha, and this gene has two promoters, the promoter 2 that is in fetal liver, pancreas, and cell culture, and promoter 1. And actually, all the cell culture have this promoter, but in the adult liver, we have always this promoter. So it's very difficult to test this in cell culture because you don't have the very important antisense. So it's very important to map those antisense and those promoters. And here comes out the idea that we need to really map all the starting site, and then actually we have been working for over the years to map initiation site with the CAGE technologies. The kappa analysis gene expression is broadly used at RIKEN, but also I know that Oleg is using very, very broadly. So, you may have heard about this, but is essentially upstream, and the transcription is from right to left here, and there is even more upstream on a different encode cell line. So you have a different initiation site. If you want to design antisense, you really need to know which cells are you analyzing. This is to design the antisense, but also to study gene regulation. This is very important, and this is a model of what is here on the right. So essentially different starting site, and there are more than 220,000 of them. And is it really important to analyze and to design synapse? And this is actually in Friedreich ataxia, and is caused by a mutation in frataxin. So there is not enough protein, and we want to use synapse to increase frataxin activity and rescue the mitochondria dysfunction. Those are all the symptoms of the disease, but we don't have the time to discuss. Anyway, this is Friedreich ataxia. And actually, we have been using CAGE at a certain point, because actually what we see is that the annotated 5' UTR in RefSeq, so in the golden standard, is actually annotated here, this RNA. And this is the first AUG. But actually, by looking at the CAGE data, there is no transcript here. The transcript starts more downstream from this and this starting site. So we can design the antisense only starting from this region. While we failed when we designed the antisense here, actually, after we verified the antisense, actually it worked very well by starting here. And actually, this is some of the data with the schematic design. The same figure we have on the genome browser now is here. And actually we have that once we put the synapse in the cells, this is the level of frataxin protein that is voters, and this is just the images of many cells where there are some fixed position here in red, but there are more variable, and each line here is a different cells. It works reasonably well, and we found that cells have a different bias for the starting site at single cell. And very important, we can identify different promoters in the control versus stimulated genes. This is peripheral broad mononuclear cells where we have we can cluster also cells that are in the control versus stimulated samples by looking at their promoter element. Importantly, actually, for everyone interested in this technology, we have been comparing ATAC-seq versus RNA expression. And our impression is that ATAC-seq identifies many sites, but by looking at RNA expression, we see that a real usage shifts. For instance, here you can see that by stimulation, we see that there is a real activity of the promoter, you can see here. But if you look at the ATAC-seq, we see that both promoters are always open and ready to transcribe, but we cannot distinguish actually the real activity. So actually what we are seeing that for identification of regulatory element in all the single cell, I think that we think that RNA expression is a very important component, actually the key component. Attack 6 is useful, but it tends to over-predict transcription starting site and so on. So with this, I'm going to just briefly close that we are analyzing different initiation site, because it is interesting for designing antisense against specific protein. But then we can also identify many different promoters and enhancers. And actually what we can study is the relationship between the nucleotide, the polymorphic sequences in those RNA and activity. So we can measure eQTL and correlate anyway expression and genotype.
Okay,
absolutely,
so
yeah.
one question is coming from me personally, actually. Yeah, we are now in Russia. We launched several big projects, and they're all based on 5' and cage for single cells. So we are so motivated for that. So thank you very much for that. So I'm interested in your claim that design is one and only RNA that's enhancing translation. Do you think it's purely because Phantom 5 is relatively shallow? Because not so super deep sequencing, and if we do deeper, if we revisit Phantom 5 samples and make 10x deeper sequencing, we will have better chances for finding these very impressive RNAs for practical use. So what would be kind of direction for this kind of hunt?
I think that this is a very good question. I think that in the Phantom 5 the sequencing was very good for the bias, because it wasn't totally non-amplified, but at the same time we sequenced only up to 5 million, 3 to 5 million per each sample. If you want to go to regular, so much per each single cell, because there is a given number of RNA, but really you need to put together cells that seem to cluster together like in some Tisney family of cells, and probably we need to sum up all the signal that comes from those cells, and that probably, I guess, we can have numbers. And actually, in the bioarchive that we have up there, I think that there is shown a little bit the way how to analyze data. That work is mostly for the analysis and how to group cells and fibroments.
And yes, and the second question comes from my collaborator, actually, because we just get funded to make kind of single cell and tissue-based AQTL-focused atlas for chickens with a different phenotypes. That's why I was so impressed that you mentioned your kind of non-human search for that kind of RNAs. So how do you think, is it a good chance to we'll be happy to collaborate, but it is a good chance to catch up the same stuff for, say, agricultural-based livestock, maybe birds, husbandry, and so on. So what are you feeling based on your preliminary study?
Well, I think that you can map a regulatory element in chicken. I had a collaborator who was at NIH, and one of his hobby was to have a company that was looking at genetics markers that improve the appetite of chicken in hot weather that usually they stop eating, and he was finding many important traits that end by of course for chicken we are not going to look at haploinsufficiencies or those diseases, but I guess that is going to be quite important to understand to understand in collaboration with the industry, I guess, to understand the traits. It's going to be also very important as a basic biology.
Please enjoy this very interesting and sophisticated talk. Thank you.
Hi everyone. Добрый день. Меня зовут Елена Гельман, и я директор Bioinformatics в Guardant Health, который находится в Redwood City, Калифорния. Я буду речь давать по-английски, потому что я здесь уже 30 лет живу, и мне так легче. So yeah, as I said, I am the director of bioinformatics at Guardant Health, a comprehensive liquid biopsy diagnostic company. And today I'll be discussing the use of comprehensive liquid biopsy in clinical trials and clinical practice. And I'll have a large emphasis on some of the analytical validation studies that we performed, which led to our FDA approval last year. I'll start with a brief background of Guardant Health and what we do. Next, I'll describe our technology and dive into one of our assays, Guardant 360, in greater detail. The bulk of my presentation will focus on our analytical validation of Guardant 360 for its FDA approval. And finally, I'll show some snapshots of how these types of tests are being used in clinical trials and clinical practice today. Guardant Health's mission is to conquer cancer with data. To do this, we have developed comprehensive liquid biopsy products across all stages of cancer and are now the market leader. We serve over 9,000 oncologists and work with over 60 biopharma partners. Importantly, we've processed samples for over 150,000 patients, and we use this massive biobank of genomic data to continuously improve our products. Our products span the continuum of care for precision oncology. Guardant 360, our FDA-approved 74-gene ctDNA panel, and Guardant Omni, our 500-gene ctDNA panel, are indicated for late-stage cancer patients to aid in treatment selection and response assessment, and also resistance monitoring across all advanced solid tumors. We also have a tissue offering in this setting. Guardant Reveal is targeted at early-stage cancer patients using a combination of genome turnaround time. We're also working on expanding our global footprint with sites in Europe, in Japan, and in China. And now I'll discuss comprehensive liquid biopsies, and in particular how our Guardant 360 test works. So liquid biopsies are a powerful tool to interrogate all aspects of a tumor in one global snapshot, assessing both inter- and intratumoral heterogeneity with one non-invasive test. Tumors slough off pieces of their DNA into circulation. However, the fraction of cell-free DNA that is tumor-derived is very small, so identifying these fragments accurately and reproducibly is a challenge, and that is why we've developed a technology platform at Guardant to overcome these hurdles and unlock cancer signals. At a high level, the Guardant workflow begins with a tube of whole blood that undergoes plasma isolation and cfDNA extraction, followed by unique molecule tagging with around 80% efficiency. Once those UMIs have been attached, we run hybrid capture to enrich the selected panel. For G360, this is a 150 kilobase gene panel. For Omni, it's a 2 megabase panel, before sequencing all of the material on a NextSeq or NovaSeq platform. And finally, the data is processed through our bioinformatics pipeline for QC and variant calling and reporting. The Guardant 360 test assays 74 cancer genes and reports SNVs, indels, copy number amplifications, fusions, and MSI. And our Guardant Omni test also offers tumor mutational burden. I'm showing here the reported analytical sensitivity and specificity of this test, but I'll go into how we calculated these metrics in greater detail in later slides. As I mentioned, the clinical Guardant workflow begins with whole blood collected in two 10 ml tubes. This is collected in Streck tubes, although we have done an equivalency study using EDTA tubes. And importantly, the minimum cfDNA input that we require for our test is just 5 nanograms of cfDNA. And you'll see why this is an important metric soon. So among the 87,293 clinical samples tested and used in this study, tested with Guardant 360 CDX or LDT of DNA input, an average of 2.94 somatic or pathogenic germline variants was reported per sample. Specifically, of the EGFR CDX variants, 26.5% were identified in samples with cfDNA yield between 5 and 15 nanograms, which could otherwise be missed if a liquid biopsy requires higher minimum input amounts. The combination of high efficiency, 80 to 90% input molecule labeling, Optimized assay conditions and hybridization probe design allows our digital sequencing platform, or referred to as DS in this figure, to recover 60 to 85% of all input molecules post-alignment, as you can see here, depending on, depending on the input amount at 5 nanograms and 30 nanograms cfDNA input. On the y-axis, you can see our molecule recovery efficiency. And this is across all types of GC content, as you can see in figure 2b, and with highly uniform coverage in figure C. In addition, our digital sequencing's in silico molecule reconstruction suppresses substitution and indel errors intrinsic to NGS workflows by more than 3 orders of magnitude relative to standard sequencing approaches. As you can see here, single-end, paired-end reads, and molecular barcoding from approximately 10 to the negative 3.5 down to 10 to the negative 6.5 with our digital sequencing error suppression modes. Now this allows accurate variant calling down to mutant allele frequencies, or MAFs, of 0.04%, or 2 molecules for certain SNVs, indels, and fusions. Scale assessment of analytical performance among several ctDNA assays. They found that detection of ctDNA variants at low MAFs below 0.5% was variable among providers. False positive rates were low, largely due to the use of UMIs, and cfDNA input was a key variable, impacting depth, sensitivity, and reproducibility. They also note that although reference samples were used in their study, they do not accurately emulate natural cfDNA features. So although we were not a part of this platform evaluation, I'll be showing similar analytical metrics performed on our platform and our validation strategy in the next few slides. Samples from 359 donors with different cancer types, as shown in the table on the right, were collected for this study, and results from Guardant 360 were compared to an externally validated NGS assay. Some important considerations. The first set of 100 samples were collected consecutively. The second set of 100 samples were selected to have the CDX variants using the comparator assay. And the third cohort was selected from the Guardant Health Biobank to include additional rare variants, including fusions. Using this data, the positive percent agreement between the 2 assays was 95 to 100% for the 3 CDX EGFR variants shown here. Panel-wide SNV PPA was 91.5%, and panel-wide indel PPA was 82.5%. Importantly, there were no MAF thresholds or cutoffs used in this study, so all variants portable CNAs and fusions was established at the lowest and highest cfDNA input amounts, 5 and 30 nanograms. For SNVs, indels, and CNAs, including our CDX variants, the LOD was established by combining cfDNA from clinical plasma samples from multiple cancers to create pools of material comprising multiple known alterations. The LOD was then established with these clinical cfDNA sample pools at 5 nanograms and 30 nanogram input, using a combination of probit and empirical approaches. Samples were titrated down at 5 different MAF values that included levels above and below the LOD, and tested across 20 replicates for 5 nanogram and 14 replicates for 30 nanogram input across the 2 reagent lots. The established LOD was confirmed for CDX variants by testing clinical patient pools exclusively from non-small cell lung cancer patients, targeting 1 to 1.5x LOD of the established LOD across at least 20 replicates at 5 nanogram inputs. And you can see here the results of the LOD study. We see that at 30 nanogram input, we have an LOD of 0.2% for each of the 3 EGFR variants with CDX claims. For fusions, where clinical samples are harder to come by, the LOD was established using cfDNA from cell lines with known fusions titrated into wild-type cfDNA from clinical plasma samples. Again, samples were titrated at 5 different MAF values for fusions across 20 replicates for 5 nanogram and 14 replicates for 30 nanogram input across 2 reagent lots. The established LOD was again confirmed using fusion-positive cfDNA. And at our lowest input amount, 5 nanograms, the median LOD for panel-wide SNVs is 1.8% MAF, and the median LOD for panel-wide indels is 2.7%. And finally, the limit of blank was established by evaluating whole blood samples from age-matched healthy donors. So 62 donor samples confirmed to be mutation negative based on sequencing with an externally validated orthogonal method were processed in the most conservative condition, using 30 nanograms of cfDNA input, with the Guardant 360 CDX across 3 lots of reagents, operator groups, and instruments. Of the 62 donor samples, 58 were tested with 4 replicates, while 4 donors were tested with 2 replicates, for a total of 240 replicates analyzed to assess the false positive rate of Guardant 360. And as you can see here in this table, we identified 0 EGFR CDX variants in all of the replicates. We found 4 samples of the 240 replicates that each contained 1 detected SNV panel-wide. 2 of the 240 samples we found an indel panel-wide, and 1 sample with a CNA call. There were no putative false positive fusions identified. And finally, importantly, the clinical data associated with our device. So the efficacy of Tagrisso relative to standard EGFR TKI therapy in patients randomized in the FLORA clinical trial, positive for EGFR exon 19 deletions or L858R mutations, assessed by tissue and by Guardant 360, or GCEAS as referred to in our SSCD, is shown in the table. The observed PFS hazard ratio of 0.41 is similar to that for the full FLORA randomized population, which shows a PFS HR of 0.46 and is referred to here as FAS. The clinical efficacy observed in the tissue and plasma positive portion of the Guardant 360 intended use population is consistent with that's found in the FAS. And the Kaplan-Meier analysis of PFS in the Guardant 360 population is shown here on the left. Low-level reversion mutations identified in 1 patient. And on the right, the hazard ratio of patients with and without reversions identified by Guardant 360. And the hazard ratio of 0.12 shows significant difference between the 2 patient populations. Another application that is gaining traction in the clinic and in clinical trials today is the use of ctDNA as a faster surrogate for molecular response. We've shown that Guardant 360 can predict responders on average 8 weeks earlier than standard RECIST. We're working through various methods of calculating this ctDNA dynamics or clearance between time points for a given patient to help identify whether they are responding to therapy or not. So once these methods are adopted more widely, patients will not have to continue on a therapy that would eventually prove ineffective. And finally, for applications such as minimum residual disease and recurrence monitoring in earlier stages of cancer, sensitivity levels are necessary that go beyond the limits of genomic profiling. In order to accurately capture even the smallest signal of tumor in circulation at early stages of disease, we are combining genomic alterations with epigenomic signatures, including methylation and fragmentomics. These orthogonal data sources are proving to be highly sensitive and specific, once trained on sufficient and relevant tumor and normal tissue-specific cohorts. And so I've discussed with you today a brief introduction of Guardant Health and the products we are developing for the clinical care continuum. We've reviewed our Guardant 360 assay in particular as the first comprehensive FDA-approved liquid biopsy and the rigorous analytical validation studies we performed for this submission. And finally, I've shown you some snapshots of how ctDNA is being used in clinical trials and clinical practice today. So with that, I'd like to thank you again for your time, and I'd be happy to answer any questions. Hopefully next year we can have this conference in person in Russia. Спасибо.
И сегодня я очень рада первый раз в своей жизни поделиться с вами своим проектом, который посвящен конкретно одному детскому раку, саркоме Юинга, и посвящен расшифровке микроокружения этой опухоли с помощью такого метода, как пространственная транскриптомика. Для начала я, наверное, хотела бы быстро рассказать, что такое саркома Юинга и почему она так интересна как модель для исследования рака. Саркома Юинга – это достаточно серьезный и очень тяжело протекающий рак, который начинается чаще всего либо в кости, либо в мягких тканях мышц, и после этого он может метастазировать практически в любой орган человека, начиная от лимфоузлов и печени, заканчивая костным мозгом и легкими. Это рак, который в большей части своей ориентирован на детей и на, как сказать, ориентирован, наверное, плохое слово, который в большей части атакует детей и молодых взрослых, подростков, и пик этого заболевания, он около 15 лет, и по данным различных клинических исследований практически нет никакой разницы между женщинами и мужчинами в заболеваемости с большим количеством инцидентов среди мужчин по сравнению с женщинами. Этот рак достаточно тяжелый, и следует отметить, что за последние 10-20 лет не было особенно никаких развитий в его терапии, и все лечение – это достаточно агрессивная химиотерапия, которая достаточно травмоопасна сама по себе. Это очень агрессивный, это очень серьезный рак, который очень быстро распространяется, и уже на момент диагностики в 20-30% случаев наблюдаются метастазы. И эти метастазы очень часто также являются устойчивыми к терапии, то есть практически всегда метастазы обозначают очень плохой исход для пациента. И саркома Юинга составляет примерно 1% от всех диагностированных раков среди детей, что достаточно большой процент, и, естественно, есть лаборатории, которые занимаются, в принципе, исключительно этим заболеванием, чтобы всего генома и всех регуляционных программ. И после этого репрограммирования запускается сет так называемых de novo энхансеров, которые активны исключительно в случае развития этого заболевания и нигде раньше, и также изменяется роль активации некоторых транскрипционных факторов, что означает, что практически при развитии саркомы Юинга происходит полное репрограммирование всей системы регуляции генов. Вдобавок к этому, к тому, что это так называемая энхансерная болезнь очень непредсказуема, она еще и очень гетерогенна. То есть то, как развивается эта болезнь, то, как происходит это репрограммирование генов, очень сильно различается как между пациентами, так и внутри самой опухоли. И таким образом очень сложно всегда понять, в каком случае какое из методов лечения будет наиболее успешным. И это все указывает на предпосылки, что необходимо продолжать заниматься клиническими исследованиями для понимания того, что же такое саркома Юинга. И сейчас я хотела бы вам представить одну очень интересную и захватывающую технологию, которая, кажется, в очередной раз произвела революцию в мире биологии. Я сейчас воспользуюсь одной аналогией, которую привела в свое время в каких-то раках крови, например, саркоме Юинга, раке кости и мышц. Почему? Потому что добавок к тому, чтобы понять, какие типы клеток там находятся, нам необходимо еще понять, как они между собой взаимодействуют, и есть ли какая-то зональность. И, продолжая нашу кулинарную аналогию, пространственную транскриптомику можно представить себе как фруктовый торт, где все не только расположено по типам, но еще имеет определенную фиксацию в пространстве. Точно так же с помощью пространственной транскриптомики можем понять не только то, какие типы клеток у нас находятся, но где именно на образце, где именно в опухоли они расположены. Всего можно выделить 3 типа методов в пространственной транскриптомике. Есть методы, основанные на методах секвенирования нового поколения. Это, соответственно, методы, где основа заключается в том, что кусочек ткани, он закрепляется на подложке, и после этого с помощью специальных баркодов мы можем распознавать каждый транскриптом в каждом так называемом споте, участке, месте на ткани, и после этого производится секвенирование. И таким образом возможно детектировать полный транскриптом какого-то определенного среза ткани очень тонкого размера. Вдобавок к этому можно делать основные методы, они основаны на анализе изображений, и тут тоже можно выделить две подгруппы. Это метод, основанный на секвенировании инсьюту, то есть каждый спот представляет собой несколько клеток вместе, но разрешение становится все выше и выше, и уже многие ученые работают над тем, чтобы делать это действительно одноклеточным, разрешение действительно на уровне каждой клетки. Это захватывающая технология, которую можно применять абсолютно везде. На самом ее начале, естественно, самые логические вопросы, которые были с помощью нее изучены, это вопрос развития, эмбриология, и после этого он уже дальше и дальше стал активно применяться в нейрологии, и сейчас в том числе при изучении различных раков и, в принципе, для изучения вопросов онкологии. В нашей лаборатории мы активно используем кит Vision, который предоставляется компанией 10x Genomics. Этот кит, он основан на работе Шталя, то есть на работе 2016 года, и это транскриптомика, которая основана на методах секвенирования. И мы заинтересованы в том, чтобы получить информацию обо всех возможных генах в нашем образце. Это очень удобная, очень интересная технология, которая, в принципе, метод я уже объяснила, и 10x предоставляет уже почти готовый кит, который позволяет выполнить все этапы подготовки образца, снятие изображения, которое после этого можно использовать для, собственно, гистологического изображения святой Анны, который обладает достаточно большим количеством замороженных образцов в их биобанке, потому что, как я уже сказала, саркома Юинга — это, соответственно, количество образцов от пациентов, которые участвовали в клинических испытаниях, оно также достаточно невысоко. В Австрии, где мы находимся, обычно детектируют 2 или 3 случая в год, не больше. И таким образом в этом биобанке нам повезло, и мы получили доступ к неплохому количеству образцов на различных стадиях заболевания, и мы выбрали 5 образцов, которые были собраны в биопсии, которые были взяты при постановке диагноза, и еще 2 при релапсе заболевания, что достаточно интересно, потому что мы хотим не только понять, как развивается этот рак в целом, но и с точки зрения концепта персонализированной медицины, понять, что является этим самым триггером, который толкает пациента к релапсу. После этого каждый из этих образцов, которые были заморожены, так как это возможно, мы секвенируем с помощью пространственной транскриптомики, и в идеале при понимании мы можем определить, какие клетки являются клетками, собственно, опухолевыми, какие клетки являются клетками микроокружения опухоли, то есть иммунными клетками, которые поддерживают ее взаимодействие, и какие клетки являются просто клетками самой ткани, в которой опухоль была детектирована. Наша задача и наша цель — это определить различные колокализированные клетки, которые взаимодействуют между собой, и те клетки опухолевого микроокружения, которые взаимодействуют с раковыми клетками. И таким образом эти клетки, если мы сможем понять, какие там существуют закономерности, мы сможем выбрать, какие из этих типов клеток могут служить потенциально нашими новыми биомаркерами или в будущем пригодятся для последующей разработки новых лекарств и новых клеточных терапий. Естественно, мы работаем над этим проектом не одни. Этот проект, как я уже упомянула, в большой степени возможен только благодаря детскому госпиталю Святой Анны и рядом находящемуся Институту исследований педиатрических раков, и в главной лаборатории Елене Тамазу, которая специализируется на биологии и на биологии именно саркомы Юинга. И комбинирование их экспертизы с экспертизой нашей лаборатории как лаборатории анализа данных и замороженными опухолевыми образцами, что гораздо сложнее. И можно также визуализировать уже на пространстве. Это я показываю вам свои опухолевые изображения из нашего исследования, насколько, в принципе, различны, но при этом достаточно информативно являются снимки этих опухолей и, соответственно, их секвенирование. И сейчас я расскажу вам самое интересное, это что же мы смогли добиться. Используя так называемую характеристическую набор генов, который был определен с помощью экспериментов клеточных линий, где был был добавлен наш онкоген, мы смогли определить набор генов, который является характерным, чья повышенная экспрессия является характерной для саркомы Ewinga, и проверить в каждом из наших спотов, в каждом из наших участков, является ли там экспрессия этого набора генов более или менее значительной по сравнению со случайной. То есть произвести такой анализ вычислительный, который позволяет нам сказать, да, эта клетка, она, скорее всего, принадлежит саркоме Ewinga, и эта клетка нет. И можно заметить, что в результате действительно заметна гетерогенность, заметен разброс, и при этом для образцов, которые были собраны при диагностике, в среднем значение этой экспрессии, этих характеристических значений, оно немножко ниже, чем для образцов, которые были собраны при релапсе, при повторном возникновении заболевания, и что представляет для нас наибольший интерес в пространстве, то есть в рамках одного образца можно также наблюдать в различных наших образцах, и опять-таки, если делать поправку на качество образцов и на то, что у нас присутствует очень высокая гетерогенность между и внутри каждого образца, все равно уже на этом этапе заметно, что в случае повторного релапса, в случае релапса опухоли, пропорциональное число клеток иммунной системы, клеток микроокружения опухоли гораздо меньше, чем в случае диагностики. И это очень интересное наблюдение, которое позволит нам в будущем выстраивать на этом основании дальше последующие гипотезы. И особенно интересно то, что, как я уже акцентировала ваше внимание на кластере иммунных клеток в этом образце, мы заметили, что в числе генов, которые экспрессируются дифференциально, то есть экспрессируются в этом участке и больше нигде, находятся два довольно интересных маркера. Один из них — это маркер системы регуляционных путей системы комплемента, который участвует в большом количестве регуляции иммунной системы, и также другой маркер, который может отвечать за развитие рака, как называется self-stemness, то есть насколько эта клетка может толкать тебя к дифференциации. То есть действительно этот участок опухоли мы смогли определить как так называемый leading edge, то есть тот участок опухоли, который продолжает расти, и это действительно интерфейс взаимодействия опухоли со всей остальной тканью. Я, наверное, остановлюсь на самом интересном месте, так как я сказала, проект еще не завершен, и это не опубликованные данные, но на данный момент я смогла показать вам, что с помощью KitaVision мы смогли произвести секвенирование достаточно большого количества большое количество замороженных образцов саркомы Ewinga, и что на пространственном разрешении...
Да, но она самая еще неизведанная. Да.
Поэтому она и неизведанная, потому что сложно.
Да. Спасибо большое. Как Наташа, вы знаете, у нас нет такого живого задавания вопросов, только через сообщение. Я спрошу, чтобы приходило. Вопрос очень хороший. Сейчас, насколько мы знаем, в России... периодически спрашиваем, раз мы существует примерно 0 мест, где можно сделать пространственную транскриптомику, и, конечно, есть коллективы, которые это хотят делать, и они всегда думают, вот с чего начинать, да? Слайд-сек пытаться адаптировать. Даже мы в ВНС и в МФТИ в сборной команде до сих пор думаем, какую систему ставить. Как вы считаете, что перспективно? Может быть, ТНХ, обкатная система?
Я хочу отметить в данный момент, что конкретно меня пригласил ТНХ. Так что и моя лаборатория является очень активными пользователями ТНХ. Наша sequencing facility делает очень много РНК-секвенирования и атак, и у нас достаточно долгая история взаимодействия с ТНХ, и мне кажется, это одна из самых продвинутых компаний. И, как я уже сказала, да, и они делают очень хорошие вещи, они делают их очень быстро и очень хорошо. И, как я уже сказала, что если интерес лаборатории заключается только в том, чтобы использовать это как метод, мы для себя решили, что нам проще использовать коммерчески доступный кит, потому что он уже доступен и он уже есть. Потому что мы не настолько заинтересованы. Например, мой проект абсолютно не заинтересован в развитии этой технологии, а в использовании этой технологии. Потому что, мне кажется, сейчас мы находимся в таком моменте, когда технологии развиваются быстрее, чем они применяются. То есть выходит то же самое например, с этой статьей про пространственную транскриптомику, которая вышла в 2016 году, и апдейты с более продвинутыми технологиями выходят год за годом, и при этом статьи, в которых этот метод на самом деле применяется для развития для исследования различных карцином и других твердых раков, появляются только сейчас с небольшим опозданием, потому что это два достаточно различных вопросов. И вегетарианский рак, и, соответственно, в этом биобанке были образцы замороженные с 80-го года, когда люди вообще еще не могли, наверное, себе представить, что будет такой метод, как пространственная транскриптомика. Но, тем не менее, эти образцы были заморожены, соответственно.
Дарья, ну вот, а как? Во всех странах врачи очень занятые люди, да? Как вот как это вот по вашим наблюдениям происходит в Австрии? С чего бы такой замечательный биобанк? Это принуждение, да, это указ? Или какое-то понимание врачей, оперирующих в том числе? Вот в чем секрет? Почему в России нет нормальных биобанков? И каждый раз такая новая история выстраивания.
Мне кажется, это очень уже политический вопрос. Единственное, что я могу сказать, что я заметила, что здесь студенты, медики, они гораздо больше задействованы в клинических исследованиях, это одно из требований. Но я не могу сказать, что в России ничего нет, так как одна из моих близких подруг, она выпускница высшей школы онкологии, и я знаю, что они тоже делают очень классные, очень крутые вещи. То есть это есть и в России, но, возможно, это не настолько близко скоординировано. Но это, опять-таки, кажется, это не работа одного года, это работа 20. То есть то, что вы сейчас пытаетесь начать делать, наверное, через 10 лет принесет плоды, и это очень классно.
То есть у вас совет, как обычно, да, все морозьте, обязательно пригодится.
Именно, да.
Это примерно вот так и работает. Все морозьте.
Но мне кажется, что на самом деле, вот я могу привести пример с высшей школы онкологии, когда моя подруга детства там училась, и сейчас она все равно с ними остается в контакте. Я восхищена тем, что они делают. Так что в России это тоже есть. Есть и врачи, заинтересованные в активных клинических исследованиях. Но, опять-таки, все постепенно приходит.
Даша, спасибо большое. Очень, очень интересная вещь. Будем на связи. Благо, что и наши проекты очень во многом завязаны с австрийскими партнерами.
Спасибо, буду рада пообщаться больше.
И даже особенно с медицинским университетом в как ни странно. Вот,
спасибо, буду рада пообщаться больше. Спасибо большое.
Да, спасибо огромное. Большое спасибо, коллеги. На этом мы нашу утреннюю дневную сессию завершаем. И у нас будет перерыв до 3 часов по Москве, до 15:00 по Москве. И у нас будет следующий очень классный набор тоже выступающих. И модерировать в основном будет Николай. Я буду на подхвате. Вот, поэтому, дорогие коллеги, спасибо за очень интересные утренние
Сразу на полный экран.
Лив, выходите из этого, Рома.
Что ж,
Все, можно. Вы в эфире.
Hi. Hi. Let me introduce another speaker. This is Dr. Piero Harchenko from Harvard Medical School, and his presentation is dedicated to transcriptional impact of disease at the single cell level.
Thank you very much. All right, so hopefully you can see my slides.
Hi. Let me introduce another speaker. This is Dr. Piero Harchenko from Harvard Medical School, and his presentation is dedicated to transcriptional impact of disease at the single cell level.
Thank you very much for the introduction. So I'll talk about much. All right, so hopefully you can see my slides.
Hi. Let me introduce another speaker.
I'm sorry, I'm getting feedback here.
Piero Harchenko from Harvard Medical School, and his presentation is dedicated to transcriptional impact of disease at the single cell.
Seems to be in a loop. Thank you very much for the introduction. So I'll talk about much. All right, so hopefully you can see my slides.
Hi. Let me introduce another speaker.
I'm sorry, I'm getting feedback here.
Piero. Yeah, it seems.
Okay, let me try. So the topic today is analysis of transcriptional impact of disease at a single cell level. And I'll talk about single cell experiments. And this is an example of a single cell experiment, although designed on proteomic data, but it's a snapshot of mouse hematopoiesis, a single experiment from Gary Nolan's group. And different cell types are colored in different ways and labeled here, but what you could see is the structure of hematopoiesis, as well as the differentiation logic. So the topic today is analysis of, again, I'm hearing myself somehow. Can you please mute? I'll talk about single cell experiments. If you're on the panel, can you please mute yourself?
Yes, 1 second, please.
Proteomic data, but it's a snapshot of mouse hematopoiesis, a single experiment from Gary Nolan's group. And different cell types. Still hearing myself. Different ways and labeled here from a minute ago. You can see is the structure of hematopoiesis, as well as the differentiation logic. So the topic today is analysis of, again, I'm hearing myself somehow. Can you please mute? I'll talk about single cell experiments. If you're on the panel, can you please mute yourself?
Yes, 1 second, please.
Proteomic data, but it's a snapshot of mouse hematopoiesis, a single experiment from Gary Nolan's group, and different cell types. Still hearing myself. Different ways and labeled here from a minute.
Yeah, I think, может быть, у одного из спикеров включен YouTube, и из-за этого дублируется, потому что у меня ничего не... Да-да.
Как бы я и вы. Окей. Так, давайте еще раз попробуем. Share screen. Okay. Yeah, so you probably heard this introduction already, but I'm going to talk about single cell analysis and analysis of disease using single cell data. So in general, single cell analysis have taken off over the past few years, and a lot of the efforts are really dedicated to sort of mapping out the base organization of the tissues. The Human Cell Atlas is sort of a landmark project in this regard, but also other initiatives to basically figure out the composition and the states of the cells that exist in the system. But the other sort of very prolific and probably insightful use of single cell techniques is in comparative setting. So in this case, we're trying to, let's say, compare a disease state with healthy state or treatment with control. And again, there are several consortia efforts that are sort of going along these lines. For instance, Human Tumor Atlas Network or Lifetime Initiative in Europe. And for this talk, I'll mostly focus on this comparative setting where we're trying to analyze disease and how disease changes the tissue. I'll, for the most part, use an example of our study from bone marrow metastasis. This is a prostate origin metastasis. So in castration-resistant prostate cancer, metastasis to the bone is nearly a universal feature that occurs in 70 to 90% of the patients. Unfortunately, there's no effective therapies at this point, and recent studies of immune checkpoint therapies have shown limited effect. It's also poorly studied tissue or samples because the material is pretty hard to get. We teamed up with surgeons to take advantage of rare spinal decompression surgeries to get fresh samples and dissociate them and perform single cell analysis on them. So here's briefly study design. So we take from each surgical case, we take 3 different fractions of the solid tumor itself, the bone marrow, liquid biopsy of the bone marrow immediately next to the site of metastasis, we call it involved bone marrow, as well as liquid biopsy further away, called distal bone marrow. As a control, we have bone marrow from patients undergoing hip replacement surgery. So they also have inflammation, but it's not cancerous in nature. And then we go through single cell analysis, and I'll try to walk you all the way to sort of validation experiments that we've done. The first thing to note, so here you have each point, let's say, sorry, each square is a different sample that we take, and each point is a cell in that sample. We have different classes of samples that we take. And the main thing I wanted to say is that, you know, in order to analyze these types of collections, you need to figure out what kinds of cell types. So, you know, this cluster may, for instance, represent T cells, and such T cells, analogous T cell states, exist in other samples, but it's not actually trivial to recognize the correspondence. And what we want to do first, before any sort of comparative analysis or expression difference analysis, is find a way to jointly track individual cell types throughout the entire collection. So if you had, let's say, a T cell population here and a T cell population here, they would actually be labeled in the same way, and you could compare them, let's say, between conditions. If you simply align data sets, you know, put matrices together, they will not align very well because each individual has its own expression pattern, its own differences, and also there are technical differences in preparation of each sample that end up putting them in a slightly different state. So the first thing that's needed is correction during alignment that allows you to establish sort of correspondence between cells. And in this case, you know, we've designed a method for doing data set alignment based on sort of multiple reflections of different samples. Essentially, we try to find, you know, best correlated cells in other sample. And since we have many samples, there are many pairwise combinations that can be done. This forms this type of a joint graph where all the cells from all the samples participate, and we can use that graph to, well, to visualize the data, but also determine these types of joint clusters that essentially persistently track individual cell types throughout the collection, and also propagate information between the samples. So this is a published work, and there are actually quite a few methods for data set alignment these days, Syrah, Harmony, and so on. All of them have their own strengths and weaknesses, and it's still work in progress, to be honest. But one way or the other, if we have this collection, and this is our bone metastasis collection, now we have sort of a joint annotation. So, all the cells, you know, let's say T cells from different samples or NK cells, actually aligned onto this position. Now, once you have that, you could start doing comparisons. For instance, here, I'm simply plotting these cells in different colors, and you have the controls, the benign samples, and all the cancer fractions are plotted in gray. And what you can see immediately is that, for instance, B cells are much more abundant in the controls than they are in cancer samples. For some reason, the generation of B cells is actually inhibited, and the amount of mature B cells in the bone marrow is decreased significantly. So this purely visual comparison, of course, you could put this on a statistical basis. So here we have a fraction of total cells in different samples. Every point is a sample, and we're simply looking for every cell type and what fraction did it account for in every sample. And here, for instance, these B cells mature and mature, and you could see that their fraction was much higher in the blue, in the benign samples and the controls compared to all the cancer samples. Now, it's a very simple analysis, but even this analysis will give you some statistically significant results in many diseases on say a dozen samples, just because the changes in the composition of the tissue can be very substantial. That analysis does have some issues, and so what we do is actually shift to a simplex space, and then this is a projection of first two principal components in that simplex space. A space that defines essentially the composition of the tissue. And what we're showing here with different colors, so the tumor and benign samples, and you can see that they separate, and the arrows show the loadings on the first two principal components. And here, for instance, these B cells that separate benign samples, and obviously tumor cells and other things separate the tumor samples. So in that space, we could do better statistics. Now, let me give you an example. This is an artificial example. So what we do is we have unperturbed sample shown in green here and perturbed sample in red. And what we do is artificially increase the proportion of cell type 1 and decrease proportion of cell type 2. So if we did this in proportions, then what you see basically is that proportions of all the other cell types are actually affected. You start seeing differences, let's say, in cell type 3, even though we didn't really change cell type 3, but our basis has changed. So what we're using is this compositional data analysis technique called CODA, and it basically runs statistical tests to determine in a more robust way what has changed in the tissue. Similarly, you could look at this as sort of a tree where the first change involves both cell type 1 and cell type 2, and then other, let's say, cell types, 3 and so on, remain unchanged. So it's basically a more robust way of analyzing proportions. This is the result for this bone marrow study. This is, for instance, difference in these B cells that I already pointed out. The tumor is much higher in the tumor fraction as you would expect, as well as macrophage populations, myeloid populations in general. This can be applied to a variety of diseases. In fact, we're doing this. So here's a very quick example from schizophrenia. Again, similar design. In this case, we're extracting nuclei in collaboration with Konstantin Khodasevich. And this is an annotation of the different cell types we see in these cortex samples. Here, again, plotting just control and schizophrenia cells with different colors, and you start seeing some kind of gradual differences, but it's hard to work out. And this is the result of this compositional analysis. In this case, the result is actually fairly simple, surprisingly so. Here I labeled all the inhibitory neurons types in blue and excitatory in red, and you could see higher proportions of excitatory neurons in schizophrenia samples for some reason. Now, this analysis that I showed you depends on the formulation of these cell types, right? And that's not always straightforward to do. You define more granular types, more coarse types, let's say group all excitatory neurons together if you want, or go in and do really detailed analysis. So to some extent, we want to make sure that we have alternatives of, let's say, compositional difference analysis that don't depend on this resolution of clustering. And here's an example. We basically use density, cell density in some kind of space. In this case, the density is calculated in this embedding space. You could also calculate it in this kind of a joint graph space. And we have control samples, schizophrenia samples. The density is calculated for every sample separately, normalized, and then added up. And once we have this, we basically have data for every point here. We have density estimates from every sample. So we can start doing statistics on this. So here we show z-scores, and high z-score, the yellow one, indicates that this population tends to be higher in schizophrenia with some statistical significance. Now, the thing to point out that some of the populations, let's say this a population of SST neurons, they're fairly uniform in terms of their behavior, so they're higher in control. Whereas other clusters like this very large L2-CUX2-M5 population actually behaves differently. So some parts of it are high in schizophrenia, some parts are high in control. And these types of cluster-free analysis allow you to check basically whether the granularity of your populations that you've defined accurately captures the behaviors that you see in the data. The other thing I wanted to describe is sort of the flip side of the compositional differences, the transcriptional differences. So you can have cell type that's present in the same abundance in both, let's say, disease and healthy state, in healthy samples, but it changes its state, right? So it changes its expression pattern, and obviously that can be very important for figuring out the mechanisms underlying the disease. So here, first, we're just going to calculate for, let's say, a pair of samples, how much did each cell type change? So we can measure some kind of expression distance between two samples for the same subpopulation. And we can do this for all the subpopulations. And the first thing we'll do is just look at this kind of weighted average, right? So it tries to estimate, the weight is according to the size of the population. So it tries to estimate the total expression differences that we see between two samples. So if we do this, we have basically distances between all the samples, right? We can embed them also into 2D space, in this case using t-SNE. So every point here is a sample, and then the colors and the shape, the colors represent different patients and the shapes represent different types. Now, you can see a few things. So all crosses, red crosses are all controls, right? So the controls cluster together, and then the cancer samples are sort of spread out around them. And the other thing you could see is that you often have a triangle and a circle that are right next to each other. And that's because this is involved and non-involved bone marrow, right? So they're very simple, similar types of samples from the same patient. And realistically, because the patient has its own expression patterns, right, these samples end up close together. Now, we could quantify this overall behavior. So here we're looking on the y-axis, the expression difference between patients, right? So every point is a pair of patients, let's say in this case, benign controls, in this case, non-involved, involved, and tumor samples themselves. And what we see is that as we sort of move towards the site of metastasis, the interpatient expression distance tends to increase, meaning that, when we get to, let's say, tumors, the patients in terms of their expression state, they start to look a lot more different than the benign controls did. Now, this is actually a trend that we see across a number of diseases. So here, going back to schizophrenia, very similar picture. The controls are much more similar to each other than the schizophrenic patients. Now, this just tells you about the distance, right? And one way, to look at that distance matrix is using some kind of an embedding. So here we use multidimensional scaling to basically project these samples into 2D space again. And what we see is that, again, the controls sort of cluster together, but the schizophrenic samples, they tend to, not go in all kinds of different directions. Now, that could have been different. They could have formed, let's say, two clusters, right? That would suggest that there might be subtypes of disease that could be followed up. But in this case, it seems to be spread out. One way or the other, these types of overviews of distances, between samples are very useful for screening the structure of the population. And here we do this using this kind of weighted total distance across all the cell types, but you could obviously do this for every individual cell type, with respect to, let's say, NK cells or myeloid cells and so on. Okay, so the transcriptional shifts, right, can also be estimated for every individual cell type, and the question they're trying to answer quite often is that, what cells are impacted the most by the disease? Because if you don't have sort of a preconceived notion, you may want to start by analyzing and following up on the most drastic changes that happen in the tissue. And so this type of ranking of cell types by the distance, the disease versus healthy distance, it becomes quite important. Now, measuring distances in high dimensional space, like between 5,000 transcripts on one side, 5,000 on the other side, is difficult. You have to reduce dimensions, you have to worry about all sorts of covariates. So what we ended up doing is actually normalizing each cell type on its own, because cell types also differ systematically in terms of their transcriptional properties, technical properties. Some have basically more mRNA molecules, and you end up having deeper coverage of these cells. So what's shown here is a standardized distance. So it's a distance between conditions, between, let's say, tumor and controls in this case, divided by the distance within conditions. So the distance is, let's say, between controls. And that gives you some kind of prioritization of cell types, in this case, progenitor cells, for instance, or PDC cells, so most changed. Again, you could have a cluster-free version of this, of expression shifts, again, to try to prioritize the areas of embedding, in this case, where the difference between disease and control appears to be most striking. And that can give you some sort of prioritization, again, on what to follow up. In the end, you have to do differential expression analysis if you're trying to interpret what is actually the nature of that expression shift, what changed between disease and control for a given cell type. And here you have a bunch of control, let's say, bunch of cells in control samples, bunch of cells in disease samples. And the method that's being used currently, and it seems to be the best one, is a pseudo-bulking method. So we basically take all the cells that we measured from a given sample, and we collapse them into essentially a bulk RNA-seq profile, right? So forget about molecular barcodes. And then your problem becomes very familiar. You have sets of bulk RNA-seq samples, right, between disease and control. And you can use familiar tools such as DSeq2 or JAR to actually run the differential expression. And that's what we use. However, it doesn't work so well in many cases. So there are limitations and one should be aware of. And the main thing is that the actual stability of these results is not great. If you have a cohort, let's say, of 10 patients and 10 controls, right, that's a sizable cohort being measured. And you could do leave one out experiments, right? So we're going to take, let's say, one of the control samples and one of the disease samples, and then rerun differential expression. And then look at stability of the results for the top 100 most differentially expressed genes. And it turns out that stability is actually quite moderate, right, or poor. 0.5 Jakarta index is not great. If you do a p-value cutoff, it becomes abysmal, right? So it doesn't work well. And we fought this for a while. It doesn't depend on the cutoff so much, or p-value or top-end genes cutoff. Improves a little bit if you get more genes. But it seems to be basically the general property of these types of tests. So here we do random group assignment, right? So this is a null hypothesis testing. And what we see essentially is that the distribution of p-values is not uniform, right? And that's supposed to be the case for a test, right? Any statistical test under null hypothesis. And we see inflation of p-values basically at the most important tail, right? Very low p-values. So this probably has to do with the assumptions that are being made by the tests, but just something to be aware of. We don't have a fix for it. Obviously, you could do overviews of what's happening. Pathway analysis is very useful. So here, for every cell type, we run either GSCA or just pathway enrichment analysis for top-end genes. And then we produce this type of summary heat map. So that means that across all these cell types, NK, MDC, and so on, you had this ER pathway, which is basically protein folding response pathway, for instance, being significantly enriched in the upregulated genes, and genes that are upregulated in involved bone marrow right next to the site of metastasis relative to controls. Same thing for downregulation. There's nothing fancy about this. It's just a convenient way of getting an overview as to what the functional changes are in the tissue. All right, so I'll just very quickly show a couple of vignettes about this particular study. So here, again, comparing the tumor samples and benign samples, we see very large differences in these myeloid populations called TIM and TAM. So these are macrophages, these are inflammatory monocytes. And then on the T cell side, right, so this is just detailed clustering of T cells. What you see is increase in these types of CTL2 populations in particular, and those tend to be exhausted subpopulations, so exhausted T cells, so they have increased expression of exhaustion markers and decreased cytotoxicity, which is a classic pattern. And so in this particular study, we thought that we could try to link the two, the appearance of these myeloid subpopulations, such as repressive macrophages, and the decrease in effectiveness of these T cells that are supposed to attack the tumor. To some extent, you could correlate them by looking across patients. So here we're looking at abundance of these repressive macrophages per tumor. And on the y-axis, we're looking at the level of exhaustion in the CTL2 population, and they do correlate, right? So that's a useful screen, especially if you have more samples. And in this case, you know, we looked at sort of potential signaling axes that could mediate this type of communication, and ended up picking a particular one. So CCL20, CCR6 seemed to be most promising, both by analysis and by the literature that was available. And so what followed then is a set of experiments to try to validate that particular axis, whether it had an impact on the T cells, their exhaustion state, and the survival overall. So we designed with our collaborators a mouse model of bone marrow metastasis in particular, a cell line that localized to the bone marrow, and then followed up with a couple of perturbations. So one was to knock out the receptor and monitor tumor growth and survival. So if we do this by simply knocking out CCR6, we see improved survival in the knockout. And the reciprocal change is to use CCL20 blocking antibody to basically interrupt this type of signaling. And again, we see improved survival in the animals when this signal is blocked. Yeah, so just quick summary. I wanted to kind of illustrate comparative study designs using single cell data. It can be more than two groups, but you know, for most of the statistical analysis, we're really focusing on one, let's say, cases versus controls. There are two kind of flip axes there. One is compositional shifts, which cell types increase or decrease in abundance. And here, you know, you have to worry about compositional analysis and effects somewhat. And the other axis is transcriptional impacts, so which cell types have changed the most. In all of these statistical tests, it's important to remember that the key difference we're trying to overcome is inter-individual variation. This is where most of the noise comes from. And when you're running a test like, let's say, differential expression test, you're mostly trying to see whether the difference overcomes this inter-individual variation. It's also important to remember that there can be all sorts of covariates and confounding factors in the design. So when you have a two-group design, you have to worry about, let's say, age of participants, you know, the conditions in which the samples were treated, and so on. Otherwise, it will confound your analysis. Okay, and with this, I just want to acknowledge my collaborators. So this was a pretty big team for the bone marrow metastasis project with hematologists, oncologists, surgeons, and a lot of postdocs. And I also wanted to acknowledge my own lab. The preprint of this bone marrow metastasis work is on bioRxiv. Thank you very much. I think you.
Ask some questions. So as far as I understood, that you have analyzed some expression levels, so mutation rates, or so on. But did you try to use gene ontology or molecular pathways? Generally, in terms of molecular pathways, if data are aggregated, the differences between healthy and disease states, between populations, between ages, are more articulated than if we took only genes, distinct genes.
Absolutely. So I kind of skipped through that screen pretty quickly, but the heat maps that I showed, the red and the blue heat maps, these were pathway analysis. And you're absolutely correct that because the data is very noisy, grouping things and testing for pathways, of course, gives you better statistical power. I should note that I talked about this problem with stability in the DE analysis currently for these relatively modest collections. So that problem actually exists for pathway analysis as well. So even though it gives us sort of better statistical power and better interpretation, of course, it does not solve the sort of bias issue that we see in the DE analysis.
But did you try some machine learning methods with blind verification, blind validation?
For DE, for differential expression?
For disease types and differential expression, for example.
Yeah, so for differential expression, most of the methods are based on kind of statistical models. So let's say negative binomial model. They fit parameters of that those distributions.
But we have some more sophisticated machine learning methods. So sometimes they are quite powerful to recognize even very slender differences, such as prognostic power for different drugs and treatment types and treatment regimens.
Yeah, I think there's space for development here. So, what we're pointing out is a limitation of the current, these popular methods anyway. And so I think going forward, it's worth looking at other alternatives.
But I'm a strong advocate for pathway analysis combined with sophisticated machine learning methods, because current bioinformatics is overflooded with some kind of dull differential analysis and p-value mining.
That could very well be. I mean, at this point, we're just trying to figure out what the sort of recommendation for general approach is. And I agree with you that that there are probably more sophisticated tools that could be tested here. But this stability criteria is, I think, one thing to keep an eye on.
Okay, thank
Peter,
you.
can I ask a very short question? Since we are projecting for, say, Russian audience at most, so a lot of people have interest of involving single-cell RNA-seq in the studies. And there is kind of very wide variety of opinions. So some people say, okay, you can use one control and one sample because so many cells inside, so they can be kind of normalizing factor. But just for you, what, like, an advice from your side.
Yeah.
Unless you can afford, say, 5 and 5 or 10 and 10 and 2 and 2, don't even think about starting because it's a waste of money. Something like this. So even short advice would be very much
so short advice. There are different purposes, right? So if you're just trying to map the composition of the tissue, you can usually get away with just 2 replicates to make sure things work, right? However, if you're doing comparative analysis, all depends on how much variation you see between your samples. So if you're working on mice, they're isogenic, they all look the same, they're all in control conditions, you don't actually need a lot of replicates, 2, 3, and you'll be fine. But if you're looking at humans, they're genetically diverse, they have different history, the environment's different, then you have to overcome that inter-individual variation. So, a few samples not going to cut it. You need, a dozen samples on each side or something close to that.
Thank you, Peter. Thank you very much.
You're welcome.
Our next speaker is Dr. Shamil Sunyaev with a presentation, Power of Numbers, Statistical Analysis of Harmonized Genomes of Patients with Undiagnosed Disease. So our next speaker is Dr. Shamil Sunyaev with a presentation, Power of Numbers, Statistical Analysis of Harmonized Genomes of Patients with Undiagnosed Diseases.
Я по-русски буду говорить, у меня.
No, but I'm afraid not everyone understands Russian in this audience. Because our audience is quite diverse and most spectators are offline and will see this transcript afterwards.
Sure. Hello. So it's a pleasure to be here and to present to this audience. So let me share my screen. Can people see my slides?
Yeah.
Perfect. So the whole story is power in numbers. The story is about harmonized data sets of human genomes, and specifically genomes of patients with rare and undiagnosed diseases. So as many people here would appreciate, the World Health Organization estimates that the collective prevalence of rare disease is quite high in the population. It's every 15th person suffers from some sort of rare condition. And while some of those, like cystic fibrosis or Duchenne dystrophy, are relatively prevalent, the vast majority are extraordinarily rare, sometimes observed in a single individual or in a single family. So the bulk of these rare conditions are represented by extraordinarily rare orphan diseases. Many of them have never been described and unknown to science. Here are some of our patients, and I should tell that all of these patients went through a diagnostic odyssey. So they were referred from one hospital to the next with a lot of unsuccessful attempts to diagnosis. Many patients do not survive until diagnosis, and their life is shorter than this diagnostic odyssey. To address this problem, the NIH-funded Undiagnosed Disease Network. So this is a network of multiple institutions that are supposed to provide diagnosis to these conditions. And I should say that most of them, not all, but the vast majority are genetic conditions. The network includes 12 clinical sites shown on the map here. It includes a sequencing core facility. It includes a model organism core. And what is important for my presentation today, it does include a coordinating center here in Boston. And all the data, especially what's important today, genomic data, are all collected at a single site at Harvard Medical School. So this is the data I will be talking about. So who are these patients? The standard process is that there is an individual who's been seen by clinicians locally, referred to another clinic, and then to the third clinic. And this is the exact diagnostic odyssey I've been talking about. So nobody can really figure out what's going on, and the disease seems to be unknown to the clinical community. So these people are referred to the Undiagnosed Disease Network to receive diagnosis. Not all of them are immediately accepted. There are criteria for acceptance. This criteria include whether diagnosis has actually been found since. We need complete medical record, very extensive. I should say that many patients now already arrive with their own sequencing of some sort complete. Then there is evaluation whether UDN workup can help, and whether patient and family are available for sequencing. Overall, over 5,000 individuals applied to the network. suffering from potentially unknown conditions, and less than 50% have been accepted so far. The conditions run a gamut of various presentations, from neurological to immune to many others. So that's not a cohort that is built around specific phenotypic presentation. Overall, about 1,600, little over 1,600 patients have been evaluated, and only quarter were diagnosed. So that's, of course, a dismal number. I should tell that a lot of this diagnoses are genetic diagnoses, while some is just pairing a patient with the right set of clinicians. So I should say that my role in this whole network is that I'm chairing bioinformatics working group that we dubbed Tool Building Coalition. So people who are building computational tools for this analysis. So what might this additional answer lie for people whose clinical record has been thoroughly evaluated? They were brought to site, there was additional clinical workup, and then full genomic screening on pretty much everybody. So what we're currently focusing on, there are unusual types of variants in known disease genes, and those would include short tandem repeats, structural variants, and non-coding variants. Also, these patients may have lesions in genes which were never characterized as disease genes. So basically, we'll focus on new gene discoveries. And in large cohorts, of course, the easiest statistical way forward is to find recurrent hits, to find multiple patients with lesions in the same gene, maybe in the same pathway or the same gene network, and try to statistically evaluate significance of these findings. Now, I should say that we're power limited because the data set is relatively small, and we're looking to work with others, and I'll talk about that a little down the road. I'll focus on two of the stories, and specifically in method development. This is not everything we do because there is a lot of activity around short tandem repeats and structural variants that I'm not going to present. Okay, the current data set is, we have, as I said, over 1,600 individuals who are very extensively clinically evaluated. We also sequence family members. So overall, it's about 4,500 genomes or exons. Now, I should say that even though the Coordinating Center has been collecting data for extensive period of time, the data are literally a mess. We have various technologies applied. They are mapped to different references, different calling tools, and different centers provide genomic data. I'm not going to talk about that. It took us 2 years to harmonize the data set. So currently we have clinical notes, phenotypic description organized in anthology, and the full jointly called data set. We started with raw reads and recalled all samples jointly together. So now the data set is complete. I should thank the department support, the SIGAP team that will present later this meeting, and also our industrial partners, Sentian, and especially Amazon Web Services. Okay, closer to the essence of the talk. The first story is about unusual types of variants, about deep intronic mutations and new open reading frames. So some mutations are seemingly non-coding, but they can affect gene product. So for example, there are splice site gains when a new site arises deep in the intron and leads to intron retention. So the transcript is completely altered. However, the mutation itself doesn't reside in the proximity to any exonic sequence and routinely missed in any analysis. A similar type of problem are alternative open reading frames when a mutations in UTR creates alternative start. And again, your transcript is being altered. I will focus on splicing today. So briefly about the analysis. So most of our cases are sporadic. meaning that we have an affected individual and all other family members are not affected. So how we can explain these sporadic cases? There's mom, there is dad. There are many variants that we don't know whether they are pathogenic, but there are many rare variants of potentially high impact on molecular function. And one option is that the individual is homozygote, so the phenotype is recessive, and we create a homozygote individual, so unfortunate individuals would inherit the same mutation from mom and dad. Now, you all were told about this in your genetic class. However, this is extraordinarily uncommon, except in specific populations with high degree of consanguinity, right? So in general population, we pretty much don't see such patients. The other mechanism is de novo mutation, meaning that mutation was not inherited from either mom or dad, but it's a new event that arose in this individual. Now, it's very difficult to deal with those because usually calls of de novo mutations are plugged by false positives, and I'll talk about the analysis of de novo mutations a little later in the talk. Another common cause, in addition to de novo mutations, is a compound heterozygosity, when two different mutations were inherited from mom and dad, but they happen to be in the same gene. So the gene is deactivated by two different mutations independently inherited from the parents. There are many combinations, so we have to prioritize those because both parents have a lot of variants, and you usually see a number of situations where two different variants were inherited in the same gene. So we need some sort of statistical framework. So of course, all interesting compound heterozygous combinations have been studied in this population. So we focused in a new type of compound heterozygosity, where one mutation resides in the coding sequence and is a high impact coding variant. But the other one is, as I just mentioned, deep intronic mutation that is either creates de novo acceptor or creates de novo donor. Right, so there is intron retention in either case, and there is change in transcript, but the mutation itself resides very deep in the intronic sequence. So the good news is that splice altering variants can be directly predicted from DNA, and we use splice AI algorithm. I'll show its performance against experimental data. We have experimental splicing reporter assays, which is multiple parallel assay, and thanks to Rich Sherwood and his lab for this collaboration. And the other reason to focus on this variant specifically is the promise of ASO therapeutics, where trying to correct splice isoform has shown amazing clinical benefit in a few rare diseases, and this is specific mutation type that is amenable to intervention with such therapeutics, unlike coding mutations. Okay, how do we compute it? So the statistical structure is relatively straightforward. So we compute tail probability of indicator variable, mutation present or absent, conditional on its score for deleteriousness, which would be various prediction scores in coding sequence and splice AI in non-coding sequence. So indicator variable if variant present or absent, and it's easy to compute what's the chance that the variant is present in the gene if you know probabilities of observing a variant in each position in the genome. So this is the relative likelihood of observing each possible rare variant. And we multiply this by the number of rare variant uniquely inherited from mom, then uniquely inherited from dad, depending on where the mutation was inherited from. And the deleteriousness score, we simply know the tail probability for splice AI score distribution. So we compute this, this probability and feed it into Poisson survival function. So we get a p-value for this event. Now, of course, the key is the analysis of relative likelihood of observing each possible rare variant per position. So we have developed, and this is primarily work of Vova Seplersky in my lab, a mutation rate map along the human genome. And I'm not going to talk about this work. That's somewhat complicated statistical statistical estimation. And now, of course, we're not dealing with de novo mutations, but we know that relative frequencies of extraordinary rare variants can be very well approximated by de novo mutational events, because forces of natural selection or biased gene conversions are negligible when we're talking about very rare events, and those are primarily very new variants. And the performance of the de novo mutation rate model on this rare variants is actually quite astounding. It predicts relative probabilities with pretty high degree of accuracy. Okay, so now we computed this score for one variant. We do absolutely analogous computation for the coding variant. And now we have to combine two p-values. And because we want both variants to be of significance, we're just taking max log p-value as our output statistic. Okay, in our data set, we have found 74 candidate genes and 30 are statistically significant after Benferroni adjustment. So how do we know that this actually works, right? That we're finding true diagnosis based on this statistical procedure. Well, first of all, we have some known diagnoses, and in this cases, most of them have been done using RNA-seq analysis. There is an advantage of our method. We do not rely on RNA-seq, and I should say that RNA-seq can work in these situations, but only for a limited subset of patients, because if your phenotype is neurological, you don't have accessible RNA from relevant tissue. We have data on diagnoses that have been made using primarily RNA-seq or just extremely fitting gene match to the phenotype. And if we sort all our cases, so every blob here represents a UDN case, if we sort according to our p-values, we see that most significant genes are enriched for true diagnosis, shown here in green, and they are completely depleted of bright red diagnoses that are definite diagnosis for a different gene, right? So we have mostly either known diagnosis, or we have new findings, or we have, I'm sorry for this, or we have very weak diagnostic hypotheses that we don't know whether they are correct or not. So the second way is we assembled expert clinical team, and they developed a metric for how much the gene fits the phenotype. They carefully analyze patient records, and this scoring scheme ranges from 5, which means it's a known well-established disease genes, there is full phenotypic concordance, to 1 where the gene doesn't seem to be functionally or genetically relevant to patient's phenotype. And we experimentally validate splice variants using massively parallel splicing assay. I should say that we're moving towards in vivo models with edited cells, but that's a longer time frame. Again, clinical team, so they consider the evidence whether a gene is known to be involved in phenotype in humans or in animals, functional and expression considerations. And we have experimental validation team that is focusing on massively parallel assays, following by in vivo models, following by the possibility of exploring therapeutics for patients. This is a plot showing mean and median difference in reads in massively parallel splicing assay for variants with different predicted splice AI scores. And as you see, all of these are positive, so on average predictions are quite okay. We observe plenty of false negatives, false positive predictions, even at high splice AI scores, but also a reasonable number of correct predictions. This is for donors, and this is the same plot this was for acceptors, and this is the same plot for donors. A couple specific diagnoses. So one of the most significant hits is gene PEX1. And PEX1 was identified with Butteroni a significant p-value, and phenotypes of the patient listed here very well fit Zellweger spectrum disorder that we believe the patient has, and our clinical team is confident enough to call it new diagnosis. Another exciting story with potentially new findings is a gene called TUF1C. And as you see, patient phenotypes involve developmental delay and intellectual disability, accompanied by heart failure, for example, and hypogonadism. And phenotype of the gene are global developmental delay, first-degree heart block, and precocious puberty. So you see that there is a pretty good phenotypic match for this gene. So we're now going through the list and analyzing this findings. Okay, so that was a story about compound heterozygotes and deep intronic splice site gains. Now I'd like to talk for a few minutes about de novo mutations. So de novo mutations, if you just take all variants with heterozygote genotype in the child and homozygote genotypes, homozygote reference genotypes in both parents, you will end up with a lot of false positives. So a couple of years ago, we wrote this package called NovaColor that works at the BAM level. And we called all de novo mutations within the UDN cohort. And at least statistically, they behave like de novo mutations. So for example, on the right here, there is correlation between number of de novo events and paternal age at birth. So we recapitulate known properties of de novo mutations, and we recovered all autosomal de novos found in initial clinical reports. So at least we know that our false negative rate is not very high. Now, one idea is just to find genes with de novo mutations happening in different patients in the same cohort. And I was very pessimistic about this approach because the cohort is quite small. Still, we use statistical machinery developed in this manuscript called de novo West that assigns statistical significance to recurrent de novo mutations in a patient panel, in a patient cohort. And we incorporated our new mutational likelihood estimates within de novo West because de novo West didn't have a sophisticated mutation rate model. So to my somewhat surprise, we found totally 9 genes with 2 or more de novo mutations within the UDN cohort, and 7 are statistically significant at Butteroni corrected level. 5 of those are already known disease genes, right? So this surprisingly works quite well, even in a small cohort of less than 2,000 patients. Of course, we're extremely power limited with this analysis. So we established collaborations with a pretty large data set from Kansas City. This is Children's Mercy Hospital. We already have all the data in-house. And other collaborators are ready to run the same scripts. And this includes Care for Rare study in Canada, Radboud University cohort in the Netherlands, and Solvar-D, which is a European cohort. We just recently reached an agreement with investigators in China, and I would be very happy to work with any similar data set in Russia. A couple stories. So this gene, CACNA1C, is the most significant gene and recapitulates to known diagnosis is basically positive control. That's an obviously correct finding of Timothy syndrome gene. Done with click of the button. And there is, this story is a little more interesting. So there are 2 individuals with de novo mutations in BAP1, and BAP1 is listed already as a diagnosis for one of them, but the other patient lists different gene as plausible diagnosis. And we're trying to figure out whether the original diagnosis was actually incorrect, and BAP1 is the cause of this patient's phenotype, or that maybe there are 2 different genetic diseases in the same individual, and they both of the genetic lesions contribute to the observed presentation. Okay, so I'm out of time. It's exactly 9 a.m. on my clock. I'd like to thank very quickly, first of all, Shilpa Kobran. So Shilpa is a postdoctoral fellow who led the study. I should say that Shilpa obtained her degree, her PhD is in computer science from Princeton. So during her postdoc, she became human geneticist. So she came with pretty much no background in human genetics. I would also like to thank Logan Blaine, Daniel Lee, and Vova Sipersky from my lab, also working in the lab Baker Rimmers, who leads the clinical team, together with Joel and Feruza here. The department, Zach, who leads the Carninci Center, Dan Travaglia was absolutely instrumental in creating the jointly called dataset alongside all other individuals, including Suzanne Churchill, who made all of this, and Kim LeBlanc, who made this organizationally possible. And of course, Rich Sherwood and his lab members, Binyap and Danielle and Tian, who helped with multiple parallel splicing assay, and all the work of the tool building coalition working group, which is our working group within Undiagnosed Disease Network. Thank you very much. I probably have maybe at most 2 minutes for questions.
Just one question. You said harmonized genomes, and what were the methods for this harmonization?
Well, harmonization is not it's a single method. So what for genomic data? So what we did, we took raw reads, and we ran joint calling process de novo, starting with of course, BWA alignment and haplotype caller, and then running them through sentient software. And this was primarily done on Amazon Cloud, creating one joint VCF, and then we use Nova caller to uniformly process de novo mutations. On the phenotypic side, phenotypes are uniformly presented as HPO terms, which is human phenotype ontology. And we also keep short PDFs of clinical notes that are a little more freehand or free text. And they are much more difficult to parse, but they are available for any individual patient analysis. Again, this is not full clinical record, but this is an abbreviated set of notes.
Can I ask a question? Shamil, it's very impressive talk. I'm surprised because I recently found that in Japan, this is also same kind of network is taking place. So it's kind of global tendency, and I'm quite impressed how you do it. But apart from splicing, so you just briefly mentioned non-coding elements in there. So we know that this is a lot of GWAS studies suggest that kind of causative variants are placed in non-coding regions. Can you say a few words about that potential? How do you feel based on your cohort? Is it true that we can expect a lot of in Hunter and promoter region, or it's overestimation?
That's an excellent question. That's an absolutely excellent question. So the question is this. Primarily, all the analysis in Mendelian genetics are focusing on coding sequence and protein coding sequence. The same actually happens in cancer genomics, and it's a complete contrast with human genetics of complex traits, where all the game is non-coding, and there is this focus on regulation. And in 2012, I think we published a paper with John Stan's group and Matt Murana saying that there is enrichment of those variants in enhancers and other regulatory regions marked by open chromatin. So there is this dichotomy. Current consensus, nobody knows, but the current consensus, I think, within Mendelian genetics is that there is small subset of mutations that are in regulatory variants, not in variants, what I mentioned, not the variants that are lie within non-coding sequence, but affect splicing, but variants that impact regulation. Nobody really knows how much. Some people believe it's 1% of the story, others believe it's 5% of the story, some believe it's 15% of the story. Pretty much everybody agrees that that's probably a minority, and this is not inconsistent with complex trait data, because in complex trait data, all these mutations have extraordinarily weak effect and never segregate in the population. So I will give you 3 vignettes why I think there is some signal there. even though I don't think that that's most of the story. I think we have a lot of undiagnosed patients. I don't think this is what explains them. But there are a few reasons we think there is signal. So reason number one is there is a paper out of DDD consortium in the UK looking at de novo mutations in known regulatory regions or regions marked by chromatin modification. They estimate that they see 2% of all pathogenic de novo mutations within these regions, and 2% is probably an underestimate. Second vignette is our own unpublished study where we focused on the same roughly method, compound heterozygosity. So we took clinical cases with known relatively common genetic disease, hearing loss, and known to be recessive, but missing the second allele. And we see enrichment of non-coding rare mutations in this cohort. So again, we estimate that some fraction are probably regulatory. We don't have any functional data on those. And the third argument is very simple. If you look at Mendelian variants in human population that do not lead to disease, but large effect allele segregating in the population, you see several examples of those in non-coding DNA. The most celebrated one is lactase persistency in lactose gene, where the causative mutation is in the intron of a neighboring gene. And the other one, which is not truly Mendelian mutation, but mutation of substantially large effect, is OCA2 for eye color. And that's a mutation within enhancers. So we do have examples of variants that segregate. Still, I don't think this story is analogous to complex disease. I think that's a minority of the story of variants.
Thank you. Very deep answer. Thank you.
Thank you very much.
Следующий спикер у нас русскоязычный доклад. Развитие симметрии, асимметрии, ранняя асимметрия развития клеточных линий выявлены с помощью мутаций. Клиника Майя. Андрей Абызов.
Алексей Абызов. Здравствуйте.
Извините, пожалуйста, Алексей Абызов.
Ничего страшного. Меня слышно хорошо, да, и экран мой видно тоже. Здравствуйте, коллеги. Сегодня я расскажу о нашей недавней работе, где мы изучали асимметрии в развитии, асимметрии в клеточных линиях. И изучали мы их с помощью мутаций. Так, как мне продвинуться дальше? Немного как бы общее вступление. Развитие многоклеточных организмов, в частности млекопитающих, начинается с одной оплодотворенной яйцеклетки и следует целому набору вполне предопределенных шагов. Но первый из них – это образование бластоцисты, потом разделение ее на внутреннюю клеточную массу и трофобласт, потом трофобласт дальше дифференцируется в два типа клеток, а внутренняя клеточная масса дифференцируется в 3 зародышевых листка и также немного вносит вклад в экстраэмбриональный эндодерм. Если посмотреть на этот процесс более детально, он представлен здесь на картине, то видно, как происходит дальнейшая дифференциация в органы и образование различных тканей организма. Справа здесь показана еще более детальная картина, где показан путь дифференциации клеток. То есть красным здесь показана зигота, далее она дифференцируется, образуются клеточные линии, и клетки специализируются в какую-то относительную, в какую-то конкретную функцию. Эта картина отображает функциональную специализацию клеток. Но параллельно с функциональной специализацией происходит еще и деление клеток, и каждый раз, когда клетки делятся, они получают мутации, то есть они их накапливаются. Существует еще процесс мутации, идет параллельно с дифференциацией. Таким образом, что если мутации возникли, скажем, поздно, например, в нейронной линии, то они будут совершенно отсутствовать там, в линии глаза или кроветворных линиях. И наоборот, если возникнут в кроветворной линии, их может быть вообще не присутствовать в нейронной линии. С другой стороны, если мутации возникли очень рано, например, там, через несколько делений после зиготы, то они могут присутствовать во всех клетках организма, конечно, во всех тканях организма, не во всех клетках, во всех тканях, с какой-то определенной частотой, достаточно может быть маленькой, но даже могут быть переданы по наследству. Вот изучение вот этого процесса образования, накопления мутаций и как это влияет на фенотип и здоровье является одним из интересов моей лаборатории. И поскольку эти два процесса идут параллельно, дифференциация и мутации, то можно использовать мутации именно для того, чтобы понимать процесс дифференцирования и образования клеточных линий. На самом деле это самый лучший способ, но он немного сложный. Каким образом мы использовали эти мутации? Мне немножко придется сделать небольшое отступление и объяснить, каким образом мутации можно обнаружить в единичных клетках и как потом это использовать. Обнаруживать мутации можно с помощью двух больших стратегий. Первая заключается в том, чтобы взять образец, и тут вот я отметил клетки разным цветом, и разный цвет соответствует, соответственно, разным мутациям. Если и вот мы видим, что в этом образце существует множество разных мутаций, которые присутствуют в клетках. Если мы возьмем первый способ, просто взять весь образец, каким-то образом его проанализировать. Ну, например, отсеквенировать и потом попытаться найти эти мутации. В чем здесь главный недостаток? В том, что когда мы находим мутации, которые очень редкие, найти сложно. Ну, потому что они очень редкие, их частота маленькая, и в данных, каких мы получаем, в любых данных, какие мы получаем, сигнал от них будет очень маленький. Это первый недостаток. Второй недостаток, что мы на самом деле не понимаем. Даже если мы нашли мутацию, нашли две мутации, мы не знаем, были ли они в одной клетке, они были в двух разных клетках. Иногда мы можем какую-то догадку сделать по их частоте, что, может быть, они были в одной. Но в большинстве случаев мы этого не можем. С другой стороны, преимущество в чем? Что у нас здесь нету никаких промежуточных шагов, поэтому если мы эти мутации, скажем, нашли, и потом подтверждаем их обратно в этом образце, это будет сразу и техническое, и биологическое подтверждение. Второй способ – это если взять образец и каким-то образом научиться извлекать или анализировать геномы одиночных клеток, единичных, и тогда потом, сравнивая, значит, мы взяли геном единичной клетки, мы его отсеквенировали, сравнили с геномом, который у нас для груды клеток, для полного образца, и тогда найти мутации. Здесь что очень важно понимать, что существует промежуточный шаг, вот этот вот, который идет, если взять клетку и получить ее геном. Как бы работать напрямую с клеткой, почти никакие технологии этого не позволяют делать, поэтому либо нужно этот геном умножать, либо клетку нужно клонировать. Поэтому есть промежуточный шаг, и поэтому важно понимать, что когда мы подтверждаем варианты, если мы их подтверждаем в ДНК, которая была умножена или клонирована, это совсем не то, что когда мы подтверждаем варианты в этом образце. То есть здесь вот небольшая разница существует. Поэтому очень важно доказать, что когда вы анализируете единичные клетки, большинство мутаций, которые вы получаете, они на самом деле нормальные. И что действительно ваш подход работает. Поэтому я несколько слайдов уделю именно этому вопросу, чтобы показать, что тот подход, который мы использовали, в частности, клонирование с помощью индуцированных плюрипатентных стволовых клеток и ПСК, что он работает. Несколько слайдов на эту тему. Работать над этим мы начали более 10 лет назад, и тогда был проект, чтобы проанализировать, каким образом фенотип аутизма, как он на молекулярном уровне появляется. И у нас был набор семей, клетки фибробластов, и из них мы выводили линии ИПСК. В то время было не очень понятно, являются ли ИПСК клонами или нет. Сейчас мы знаем, что они, да, являются клонами, и вот следующих несколько слайдов именно это вам покажут. Вот, а поскольку они являются клонами, то есть у нас ситуация вот такая, что если здесь есть мутация, то мы получили клон, и все клетки в этой колонии будут иметь эту мутацию, соответственно, в другой колонии будет другая мутация, и сравнивая их между собой, мы эти мутации можем находить. Но начнем с того, как доказать, что действительно ситуация такая. Мы проанализировали ИПСК порядка 10 лет назад. Идея была в том, чтобы посмотреть, есть ли у них какие-нибудь большие геномные перестановки. Оказалось, что действительно они бывают. И вот здесь показано следующее. Значит, это фибробласты, образец фибробластов, который как бы изначальный оригинальный образец. И вот это 3 ИПСК-линии, которые были выведены из этих фибробластов. Значит, по оси это геномная координата на 10-й хромосоме. А черным цветом показано количество ридов, то есть мы отсеквенировали фибробласты и ИПСК, и количество ридов, которые были раскартированы на геном в определенных бинах, то есть ячейках генома. Вот. А зеленым это показана сегментация вот этого черного сигнала уже с помощью аналитической программы, которую мы разработали. Ну и видно, что в 2 ИПСК-линиях ничего не происходит, то есть сигнал абсолютно ровный, точно так же, как в фибробластах. Ну а в последней, здесь вот 2 района отмечены, что там количество ридов больше, и приблизительно больше на 50%. То есть что это означает, что там была дупликация. То есть эти районы были дуплицированы. Ну и, естественно, как бы вопрос возникает, почему они были дуплицированы. Это результат того, что произошло перепрограммирование, и каким-то образом они там возникли, либо это результат того, что они являются клональными. Вот. То есть сейчас мы знаем, что ситуация именно такая, и доказательством этому являются следующие. В одном из образцов фибробластов у нас было 2 ИПСК-линии, где существовала дупликация. Вот, и дупликация была практически та же самая, то есть координаты ее были очень близки друг к другу. Вот, тогда мы предположили следующую ситуацию, что у нас уже в фибробластах существует набор клеток, которые без дупликации, и клетки, которые с дупликацией. Когда мы производим клонирование, просто случайным образом у нас так получилось, что 2 колонии, у них была дупликация, и она теперь присутствует во всех клетках этой колонии, этого клона. Вот. Чтобы доказать это, мы делали простой ПЦР-анализ. ПЦР запустили на точке перестановки вот этой дупликации, там, где у нас есть точка связки двух копий. И у нас был сигнал в ИПСК номер 3, она вот эта вот ПСК номер 4, не было ее в ИПСК номер 1, не было в контроле. Но если мы сделаем фибробласты, то мы увидим там тоже сигнал, то есть диагностическую полоску. Вот. Более того, что это классический ПЦР, можно использовать количественный ПЦР, можно в этом количественном ПЦР посмотреть, какая частота вот этой мутации. Значит, идея как бы ПЦР та же самая, единственное, что она делается параллельно в 1000 микрокапель, или даже несколько десятков тысяч микрокапель. Вот здесь вот 2 сигнала. Значит, это сигнал для дупликации, а это сигнал для контрольного аллеля, чтобы сделать нормализацию. Ну и в итоге, если мы как бы сделаем эти эксперименты, если мы перенормируем частоту этих 2 аллелей, в итоге получается, что в фибробластах это порядка 100%, клеток имеют дупликат, извиняюсь, не в фибробластах, а в ИПСК-линиях в 3-4 это 100%, а в фибробластах это порядка 12,6%. То есть мы абсолютно точно можем доказать, что действительно вот эти вот дупликации, иногда делиции мы тоже видим, в ИПСК, они присутствовали в фибробластах. То есть это в этом одном конкретном случае. И не только мы можем доказать, мы даже можем увидеть частоту, какая была в фибробластах. В данном случае 12,6% клеток. Ну, дальше мы сделали несколько таких экспериментов, и в целом мы показали, что порядка половина таких вот больших вариаций числа копий в ИПСК, они могут быть отслежены как бы в изначальном образце найдены. Только половина, вопрос в чем, почему только половина? Ответов может быть несколько, может быть действительно часть из этих больших перестановок возникли в результате перепрограммирования или клонирования ИПСК-линий. Это один из вариантов. Второй вариант, мы считаем, что более вероятный, это то, что то, что часть из вот этих вот перестановок, она настолько редкая в фибробластах, что мы не можем ее увидеть даже с очень чувствительными технологиями, такими как количественный ПЦР. То есть мы оценили, что наша чувствительность, она до порядка 1,1%. 0,1%. То есть если они присутствуют гораздо реже в фибробластах, мы их не сможем увидеть. Ну, экстремальный случай, например, что вот такая дупликация или делиция присутствовала в одной единственной клетке фибробласта, и эту единственную клетку мы клонировали и получили из нее ИПСК, то есть, соответственно, все вариации, какие мы потом найдем, если мы вернемся в начальный образец, то мы не сможем подтвердить, что в фибробластах эта мутация когда-то была, потому что она теперь только присутствует в ИПСК-линиях. Вот. Но, с другой стороны, 50% это уже много, показывает, что действительно, скорее всего, все, что мы видим в ИПСК-линиях, это действительно мутации, изначально присутствующие в фибробластах. Я тут слежу за временем. Вот. Ну и второе, мы оценили, что порядка 30% клеток фибробластов имеют вариации в числе копий, то есть это очень много, большой процент. Далее, что мы сделали, мы посмотрели на точечные мутации. Точечные мутации немного сложнее находить и подтверждать по той причине, что когда вы исследуете большие перестановки, вы можете интегрировать сигнал с достаточно большого интервала в вашем геноме, и вы можете видеть разницу, и сигнал более четкий. Когда же вы используете и смотрите на одну мутацию, то есть это одно отличие одной базы из порядка там нескольких сотен баз, если вы там делаете ПЦР, если вы делаете обычное секвенирование. Вот. Это немножко сложнее, но мы потратили на это достаточно много времени и в итоге подтвердили, что действительно даже точечные мутации можно находить с помощью клонирования. Вот это один из слайдов, демонстрирующий это мутации, которые мы нашли с помощью ИПСК-линий, а потом что мы сделали? Мы просто сделали очень глубокое секвенирование культуры клеток фибробластов, которые мы использовали. Вот. И что здесь показано, что вот красным, вот справа, значит, это частота мутаций в фибробластах, точечных мутаций. Часть из них были унаследуемые в красном, то есть это была наша ошибка при нахождении. Ну а вот, например, зеленые это были мозаичные мутации. Их было достаточно много, и видно, что они образовывали такие вот пики, как бы горочки в распределении, причем даже существовал зазор между ними. То есть мы это объясняем тем, что существовалась клональность. Именно в том образце кожи, который был взят у конкретного человека, там существовала клональность, что большинство клеток были клонами какой-то одной клеточной линии. Вот. Ну и дополнительно, что мы подтвердили, что именно в клонах самих, в их ПСК, частота мутаций порядка 50%. То есть теперь у нас как бы есть экспериментальная система, с помощью которой мы можем достаточно очень точно и уверенно исследовать мозаичные мутации на уровне единичной клетки. То есть мы берем клетку, клонируем, потом с помощью ИПСК секвенируем и можем смотреть, что происходит в одной конкретной клетке. Ну а теперь мы это можем использовать для разных идей, там, например, смотреть мутационную нагрузку, смотреть, как это меняется с возрастом. Но сегодня я буду рассказывать немножко о том, как мы посмотрели, как можно смотреть историю развития организма с помощью мутаций. То есть здесь показана общая идея того, как это выполняется. Если мы хотим отследить клеточную линию, скажем, в модельном организме, ну, например, мыши, то мы можем просто сделать, обычно это делается с помощью внесения геномных изменений, трансгенная мышь, например, ввести сюда зеленый флуоресцентный белок, и он в какой-то момент будет активирован, и тогда мы можем, ну, например, увидеть даже на коже вот какую-то область, которая там светится, или ну, каким-то подобным образом. Не обязательно это будет видно прямо вот так вот на коже, там можно с помощью других исследований, но в принципе это все делается именно таким образом. Естественно, что применить к людям такой подход мы не можем, потому что это этически неправильно, мы не можем делать эксперименты с людьми, делать генетические мутации, генетические изменения. Вот. Но что мы можем? Мы можем... Зная то, что практически при каждом делении мы получаем новую мутацию, мы знаем, что у нас клетки, они накапливают очень много мутаций. То есть, в частности, вот мы видим, что, кстати, извините, я забыл сказать, что когда мы изучали фибробласты точные мутации, было порядка 1000 мутаций на одну клетку, то есть огромное количество. Вот. гораздо больше, чем оцененное количество делений. То есть деления считаются порядка там 30 до развития, потом они потихоньку делятся еще в течение жизни, а мутаций у нас там сотни тысячи. То есть мутаций очень много. И вот если у нас происходит эмбриональное развитие, вот это у нас зигота, то при первом делении, скажем, ну условно говоря, первые две клетки, первые два бластомера получили мутацию альфа-1, альфа-2, потом дальше делятся, получают мутации бета, потом получают мутации гамма. То есть и вот если взять вот эту клетку, я ее покрасил оранжевым цветом, то и все эти клетки, они будут унаследованы последователями. Все эти мутации будут унаследованы последователями вот этой клетки. То есть может быть такая область, скажем, в коже или в органе, где у нас просто какой-то определенный набор мутаций у большинства клеток. То есть не обязательно это будет видно, вот, но если мы эти клетки проанализируем, и потом, если мы проанализируем достаточно большое количество клеток, то по общим мутациям мы можем восстановить хронологию, как клетки делились друг от друга, и восстановить эту историю развития. Это то, чем мы занимались. Вот. И, значит, дизайн нашей работы был следующим. Мы взяли человека, живущего, это был больной синдромом Туретта, мы взяли 6 биопсий кожных фибробластов. Из них мы сделали 74 ИПСК-линии, обнаружили мутации, сравнивая их друг с другом. То есть вот тут могут быть, например, зеленая линия, зеленая мутация, красная с красной. Сравнивая друг с другом, мы их можем найти. Вот. Еще дополнительно, что мы сделали, мы отсеквенировали кровь, слюну и мочу из пациента с большим покрытием. Если мы секвенируем ИПСК-линии с покрытием 30Х, то здесь кровь, слюна, моча была с покрытием 200Х. Вот. И потом, объединяя все эти данные, мы вот именно занимались тем, что восстанавливали вот это вот клеточное дерево клеточных линий с раннего развития. Вот такое дерево у нас получилось. Это достаточно сложная картинка. Я потрачу некоторое время, чтобы объяснить, что здесь нарисовано. Значит, вот это вот зигота, потом вот это клеточные линии, значит, после первого деления. Значит, буквами указаны именно мутации, которые возникли при этом делении. Вот здесь, вот в этом бластомере 4 мутации A, B, C, D, а здесь пока ни одной мутации нету. На самом деле мы потом нашли одну, но пока еще не опубликовали это. Значит, здесь вот в этой области мы указываем, в этой ветви мы указываем мутации латинскими буквами, в нижнем мы указываем греческими буквами. Но еще там, где есть мутации, мы показываем их частоту в крови, слюне и моче. Вот. Что касается вот этих квадратиков посередине, на самом деле это важный параметр. Сейчас объясню, что это такое. И еще было несколько мутаций, которые мы нашли в тканях, но мы их не нашли с помощью ИПСК-линий. То есть, скорее всего, просто мы еще недостаточно много ИПСК-линий сделали для этого пациента. Так, что же были эти квадратики? Тут эти квадратики показывают нам о том, насколько полностью мы восстанавливаем деление, каждое деление. То есть идея очень простая. Если у нас есть зигота, и она делится на 2 бластомера, и пусть частота одного бластомера будет F1, а другого будет F2. Это частота в каком-то конкретном образце. Образец может быть вообще все, что угодно. Может быть слюна, моча, кровь, фибробласты, неважно. Главное, чтобы это было в одной ткани. Частота F1, F2, то тогда F1 плюс F2 будет всегда 100% в любой ткани. F1 и F2 может конкретно меняться в каждой ткани в зависимости от того, как произошло развитие, но всегда сумма их должна быть равна 100%. То есть тогда, если мы отложим F1 и F2, если, допустим, у нас есть 2 мутации, или, там, которые у нас маркируют 2 линии, и мы считаем, что F1 и F2 – это их частота, то тогда их сумма должна всегда быть 100%, то есть откладывая их по 2 осям, мы должны всегда получать точки, лежащие на диагонали. Вот тут вот пример. Например, кровь, моча и слюна, то есть они должны быть на этой диагонали. При этом, если у нас вклад равномерный каждой из линий, то они должны быть приблизительно по 50% в каждом образце, то есть должны быть здесь. Ну, тут пример показан, что они отклоняются от этого. Лежат точно на диагонали, но отклоняются от равного вклада. Вот, соответственно, если потом у нас происходит последующее деление, то тогда частота 2 дочерних клеток или линий F1' плюс F2' будет равна F0, это частота материнской клетки линий. И тогда, если мы поделим все это на F0, мы получаем то же самое уравнение и получаем ту же самую картинку. Вот, поэтому что что является полным делением? Является полным делением, если во многих образцах мы видим, что точки расположены строго на диагонали. Это мы и должны смотреть. Ну и вот первое деление, мы видим, что у нас 3 образца, слюна, моча и кровь, и действительно частоты вот двух вот этих вот ветвей, они расположены строго на диагонали, то есть что дает нам уверенность в том, что вот это вот деление было восстановлено полностью и с большой точностью. Ну вот здесь вот деление тоже приблизительно на диагонали. Вот здесь вот деление, некоторые образцы отклоняются от диагонали, то есть, скорее всего, не было полностью восстановлено. То есть, наверное, здесь были еще какие-то деления, но мы не все из них видим и так далее. То есть мы уверены в том, что, по крайней мере, несколько делений мы полностью восстановили. Вот, и наблюдаем вот такую вот картину. Значит, это дерево, оно, конечно, очень длинное. Вот это вот клеточная линия, те, которые мы сейчас измеряем, которые в ПСК получаются. Вот, а это то, что было в начале при делении. Что, во-первых, заметно, сразу бросается в глаза, что вот уже при первом делении вот эти вот 2 ветви, они вносят совершенно неравномерный вклад. То есть вклад вот этой линии, он намного больше количество клеток в организме, чем вклад вот этой линии. Поэтому мы, соответственно, назвали это доминантной, это рецессивной. То есть в крови вот эта доминантная клеточная линия 90%, 91% клеток образует. А в слюне чуть поменьше, в моче еще чуть поменьше, но в принципе тенденция однозначна. Вот. Естественно, нас это очень сильно удивило. То есть это было как бы, ну, скажем так, даже шокирующе. Поэтому мы решили, что, может быть, это больной с синдромом Туретта, может быть, это как-то связано с его болезнью, тем более, что у него симптомы были несимметричные. Поэтому мы решили взять условно здорового человека, посмотреть, что там получится. И отсеквенировали мы меньше линий, но в итоге пронаблюдали абсолютно тот же самый результат. Вот это вот первое деление. Опять все, значит, частоты у нас лежат на диагонали, и опять одна линия, она доминирует, а другая линия, она как бы рецессивная. Вот, но здесь что было интересно, что у рецессивной линии уже прямо при первом делении у нас огромное количество мутаций, то есть порядка 10, 2 индела и еще порядка 10 точечных мутаций, что было как бы, ну, немножко отличалось от больного Туретом. Вот. И тогда мы проанализировали еще данные, которые мы в предыдущей нашей работе, когда мы изучали мутации в эмбриональном мозгу, мы нашли еще дополнительно несколько мутаций, несколько инделей, вот, и восстановили клеточное дерево вот этих клеточных линий в этом же мозгу. Вот. И в принципе в эмбриональном мозгу, то есть вот это вот дерево, которое мы построили, мы не видели больших отклонений от симметрии. То есть вроде как одна линия тоже доминировала над другой, но как бы количество было не так сильно отличалось по количеству клеток. Асимметрия была небольшая. Но здесь я бы хотел подчеркнуть один важный момент, что в частности вот в этой ветви здесь нету мутации вообще. То есть здесь вот есть одна мутация, а здесь этих мутаций вообще нету. Это создает некую неоднозначность. То есть если вот у нас мы можем дерево сделать таким образом, да, а мы можем, поскольку здесь мутации нет, мы можем соединить вот эту ветвь с мутацией A, с мутацией альфа, или мы можем соединить вот эту вот ветвь с большим количеством мутаций тоже с ветвью, которая альфа. То есть у нас возникают различные варианты, и без дополнительных мутаций, которые мы можем найти, мы не можем разделить, какой вариант правильный, какой нет. Но, то есть вот это вот 3 разных варианта. Но вот это вот один из вариантов, который в середине, то здесь видно, что если такое дерево, если оно может быть правильное, то количество мутаций здесь у рецессивной линии, то есть существует опять тоже рецессивная и доминантная линия, и очень большое количество мутаций у рецессивной линии, то есть абсолютно точно так же. Да?
Следующий спикер уже ждет. То есть пора кончать.
Хорошо, да. Я сейчас быстро закончу. То есть здесь у них количество мутаций такое же большое в рецессивной линии, как и у условно здорового человека. Так, ну а что касается больного с синдромом Туретта, у него вроде бы в рецессивной линии не было мутаций, но мы обнаружили большое количество инделей в разных большое количество индолей в последующих делений, и это статистически значимый результат. То есть сейчас было несколько работ, которые были опубликованы до нашей работы и после нашей работы, которые занимались подобными вещами. Вот работы из 2018 года. Здесь они не видели такой большой симметрии, как у нас. Но вот последующие их работы, действительно уже опубликованные после нашей работы, тоже видели большие асимметрии, доминантную рецессивную линию. Вот, и тоже с большим количеством мутаций в рецессивной линии. То есть результаты вроде как более-менее сходятся по группам. То есть не во всех 100% наблюдается такая большая симметрия, но она наблюдается разными группами. И в частности, вот тоже какие? В этой работе. Вот, выводы сейчас, 2 слайда буквально. Возможно, объяснение следующее. Поскольку мы видим, что у нас существует большое количество мутаций, один из возможных выводов, что поскольку клетки могут неравномерно делиться, то есть в доминантной рецессивной линии, доминантная рецессивная линия, может быть, делится медленнее, поэтому, когда мы производим ретроспективную реконструкцию, то нам кажется, что это было 2 деления. На самом деле, поскольку временной компонент мы не можем отследить, то есть существует отличие между 2 линиями. А второе возможное объяснение, что у рецессивной линии каким-то образом клетки, например, либо умирают более часто, либо они вносят вклад в плаценту, в экстраэмбриональную, которую мы у живых людей, соответственно, у нас нет, и мы не можем про это пронаблюдать. Ну и, например, вот такая вот интерпретация, возможно, скажем так, в научном мире существуют споры по поводу того, правда это или нет, но в частности вот такая вот интерпретация, что начиная с зиготы клетки уже неравномерные, и одна клетка как бы осуществляет, вносит вклад в развитие живого организма, а другая в плаценту. Может быть, это объяснение того, что мы наблюдаем. Но я тут пропущу несколько слайдов и пойду сразу к выводам, что послезиготные мутации чрезвычайно распространены в клетках, что одна из двухклеточных линий доминирует уже после первого дробления. Может быть, качество репарации влияет фактор. И вот последний вывод, который я напрямую не говорил об этом, что тот метод, который мы разработали, он является минимально инвазивным, и то, в принципе, его можно использовать для реконструкции эмбрионального развития в каждом живом человеке. Ну, на этом я закончу, и спасибо за внимание, это благодарность.
И, вот такой вопрос сразу после доклада. Что я слышал от многих авторов, что по типу мутации можно судить, какая-то мутация герминальная или соматическая. Это подтверждается?
Это в некоторых случаях да, в некоторых нет. То есть если мы говорим о ранних мутациях, то там по типу мутации нельзя судить, потому что они, грубо говоря, герминальные и соматические, они одинаковые. А впоследствии потом уже мутации, которые соматически возникают под воздействием среды, ну, например, ультрафиолета, то там уже есть большие отличия.
Спасибо.
Пожалуйста. Мне уже тогда отключать свой экран?
Да.
Угу.
Next speaker is Dr. Benno Graf from Memorial University of Newfoundland, decoding uncharted genomic variation in acute myeloid leukemia using long-read sequencing technology.
Okay, thank you, Nicola, for this presentation. Can all you guys see my screen?
Yeah.
Okay, so as you mentioned, my name is Benno Graf. I am an assistant professor at Canada Research Chair in Bioinformatics for Personalized Medicines at the University of Memorial University in Newfoundland. And I am also a visiting assistant professor at the Cancer Science Institute of Singapore. So the work that I'm going to present to you today, it's a work in collaboration with both universities, which involve people from Newfoundland and also people from Singapore. So most of this work actually has been done by my PhD student, Tam Chen Yong, so from Singapore. And this work actually also was taken over with one of our engineers here in Newfoundland named Geoff Woodland. So what I'm going to show you today is how we can use the latest technology of sequencer, so we call the third generation of sequencer, to find novel structural variants which were not detectable with previous technology. So the first questions I want to ask you is, what is NGS? What means NGS? And most of the time, people will answer NGS means next generation sequencing. But next to what? And actually, it's next to the Sanger sequencing, which was the first generation of sequencer. So next generation sequencing is actually the second generation of sequencer. So what I'm going to talk today is the next generation of sequencer, which is the third generation of sequencer. So just to give you an overview of what we call next generation of sequencer, just to give you an update, is all the sequencer that provide high throughput sequencing, but in a short reads. And what you may have in your laboratory is either the ion torrent sequencer, and all the Illumina's sequencer belong to the second generation of sequencer, so from the mini-seq to the NovaSeq. So here, for instance, in Newfoundland, we use actually actively the MiSeq, the mini-seq, and the NovaSeq, same in Singapore. And this actually a sequencer that we call second generation of sequencer. So there were actually a few generations of sequencer, as I mentioned. So the first one was the Sanger style, so we call that the classic sequencer. Then the first next generation of sequencer was the Roche 454, and then come the solid ABA Solid, and what we call the next gens, and also the Helicos. So, for instance, in Japan, the RIKEN Institute a long time ago, they use a lot the Helicos, which was a bit more advanced, and that's why we call that the 2.5 generations. But unfortunately, both of the sequencers now disappear, and Illumina now trust the market. And now we come to the third generation of sequencer with two brands, so the PacBio and the Nanopore. So this is what I call next generation, so third generation of sequencer. So quick update about the second generation of sequencer. So it produces short reads, piece of DNA that runs from 75 to 250 base pair in a high throughput. So depending which sequencer you are going to use, you can throughput 1 million to 5 billion of this small fragment of DNA. And the way that it works is that it does not actually sequence the entire fragment that you put in the library, but it sequences only the extremity of this fragment. So most of the time we optimize the library, the DNA library to range to be around 500 base pair. And what the sequencer is doing is just sequence the first 150 base pair, or I mean, depending what library you use, on both sides of your DNA strand. And then the next stage is to align this read. So this is different. Fragment is what you give to the sequencer, and read is what the sequencer gives to you, is just the extremity. These reads are mapped to a reference genomes to find the correct locations. And then we can actually, when you map all the reads, this is how you rebuild the puzzle. So sequencing using this technology is actually cutting the genome and rebuild a huge puzzle. So if we want to do that very, very properly, it's really computationally demanding because it's actually complicated, and particularly when you have repetitive elements. So imagine that you have a puzzle with the same shape, same color. So it's really difficult to find where this puzzle, where this piece actually goes into this puzzle. And also it's rely on our reference genomes. And you will see that the reference genomes that we use is not actually a reference genomes that is applicable to all ethnical groups, to all individuals, because it was mainly built on Caucasian genomes. And we do see that between individual, and this is what we see when we study Asian genomes, that we find a lot of structural variants that are not pathogenic, but just polymorphic, which belong to the ethnical group. So very quickly, the way that second generation works is required, as I said, reference genomes. And the protocol is that you have your individual genome, you fragment these genomes, then you give this fragment to the sequencer. The sequencer identify each nucleotide within this fragment, and then you align that to reference genomes. So when you find a read with a mismatch, we consider that as a mutation. And for instance, in this mismatch, you have in genome reference, you have a C, here you have a G, and that is considered as mutation G2C. So when you have a short read, so for the second generation sequencer, what you need to do is to reconstruct a very complicated puzzle. With the third generation of sequencer, we do have to reconstruct a puzzle, but with less pieces, and which make much more easier to identify the differences between the reference genomes and the genome of interest. And also we can rebuild a genome of reference much more accurate. So unfortunately, the third generation sequencer has also some bias, and here I'm going to show you some differences between second generation and third generation sequencer. So to date, the third generation is not able actually to replace fully the second generations, and we still need both for many reasons. So first of all, the second generations can provide very high throughput reads, but the drawback is that it's short fragments. But one big advantage of the second generation sequencer is that this is the most accurate technology so far. And as I said, it's very high throughput for very cheap. Whereas the main advantage of the third generation is that it can actually throughput very long reads. So instead of 100 base pair, you can go to kilobase. So for instance, our protocol, we do an average between 6 kb to 10 kb, but the longest that we were able to do in our lab was 400 kb. And you can see on the publication, some people actually were able to sequence much longer than that. The drawback of this technology is that there is a low accuracy, and although the chemistry keep improve, this is nothing comparable to what we have with the second generation of sequencer, where the accuracy is close to 100%. And then we can have a similar genome coverage, but with a lower sequencing depth, which means that if we want to have the same sequencing depth as a second generation of sequencer, we need to pay much more. So the price is per kilobase sequencing is much more higher. So now I'm going to give you an introduction to genetic variations. So we have actually several categories of genetic variations in our in my presentation. So I'm going to present you what is structural variant. So I'm going to focus on the structural variant, but I just wanted to positionate structural variant with other genetic variations. So the one that everyone actually is used to see is the single nucleotide variations, or SNV. This is actually just a single nucleotide that changed from the reference genome to the individual which has been sequenced. So it's only one base pair. We see that every 4 to 5 million per genome, and the SNP frequency per genome is a bit more than 1%. Then we have what we call the small insertions or deletions, or in short, indel. Indel, actually, the definition is everything which is insert or removed from the in a genome, in an individual genome. So for instance, the individual has 4 base pair deletions here, which is actually in a reference genome. The canonical definitions of insert is everything which is remove or insert, which range from 1 base pair to 49 base pair. These events appear every 700 kb to 100 kb per genome. So you see it's a bit more frequent than that. And the last one is the structural variant. So it's a huge family which include big deletion. So everything which is more than 49 base pair, so greater than 50 base pair. It can be also insertion. So it include big indel, in other words, but not only, also duplication. So duplications actually is the insertions of one specific piece of the same genome. So the genome is duplicated, but also inversions and translocations. And very surprisingly, the latest report published during the last few years, thanks to the third generation of sequencing, found that these kind of events occurs in average every 23 kb to 28 kb per genomes. So you can see that structural variants is a genomic events that is more occur more often than the indels and the mutations. So this is one of the reasons we were extremely interested in investigating that in disease that are not known to have a lot of mutations, and we wanted to know if structural variants can play a role in several disorders. So, in summary, SNV outnumber structural variant outnumber the SNV, so from 3.4 to 1 in terms of base pair. Structural variants have a larger area of impact in the genomes, and structural variants are less characterized than the SNP SNVs, mostly due to the limit and the constraint of the previous technology. So to support that, I just wanted to show you a few publications. So one of them is a publication published in Cells in 2019 when they sequence different ethnical groups. And what they show over here is that you can see in this bar chart all the the structural variants across the patients. So when you compare that to the genome reference, and you can see that just a few are shared across all the individual. Some of them in purple are a major allele, which means that they are present in more than 50% of all the samples, but not all. Then you can see in blue here, some of them are polymorphics. That means that they are present in less than 50%. And some of them are unique, so in light blue, unique to the individual. And you can see that the ones that are more unique here, these two individuals, you see much more structural variants specific to this individual are from African descendants. And that actually is due to the fact that the genome reference that we use to assess the structural variant here is mostly done by using Caucasian genome. So, and it does not take into account other ethnical groups, and we need to have actually other filter here to segregate structural variants that are that belong to the ethnical group from structural variants that are purely pathogenic. So we know that structural variant is already involved in many diseases. So this is the case of the Down syndrome, the Turner syndrome, few Mendelian diseases, a neurological disorder, and also a lot of different cancers. And to date, the consequence of the structural variants actually are used for targeted therapy, so for personalized medicines. And this is a case for gene fusion, so the BCR-ABL in CML. So this is a kind of leukemia where we use the amitriptyline drugs. So same when we have this gene application, so RBT2 in breast cancer. So we use actually a specific drug for that. So when we are able to detect that. And BRCA1 in breast cancer as well. So where we were able to find pathological disorder at the structural variant level, in some cases, we were able actually to design some drugs. And the aim of the tools that we are developing and the aim of the work of my team is to further discover new structural variants in other diseases. So we take as an example the AML, so acute myeloid leukemia, for one main reason. So it's because in Singapore, this is actually a disease that affects a lot of patients. And you can see here, half of the patients are diagnosed as a normal karyotype. So which means that when these individual patients go to the clinics, when we do a sequencing or when we sequence the genome, we cannot see any abnormality in the genomes. So and the other half, actually, you can see a few of them have inversions, translocations, etc., etc. So what we did is we took this portion, so almost 50% of the patients, and we resequenced them to see if we were able to find structural variants using the third generation technology of sequencer. In other words, what we wanted to do is to see if the fact that we didn't find anything here at the previous stage is just because of the limitations of the technology and not because the genomes of these patients are normal. And this is what we did is we sequenced these patients with both technology, Illumina with second generation sequencing and Nanopore with with sequencing, so third generation of sequencing. And we combined both. So basically what we did with the long reads, we wanted also to find insertions, duplication, translocation, deletions, and inversions, everything which is larger than 50 base pair. And we know that the limitations of that is the high error rate, the lower throughput, and the high cost of sequencing by base pair. So right now, what I'm going to explain you is the result of our discovery, is that we have assessed the MinION sequencer from Oxford Nanopore for whole genome sequencing in a human and particularly in patients. We have developed a reliable and low depth long read variant coding as a bioinformatics tool that lower the cost of the sequencing for Oxford Nanopore sequencers, but it works also on the PacBio. And we have identified an AML associated variant in a clinical sample that we are currently investigating. So basically, one of the reasons we reassess the accuracy of Nanopore is because the previous assessment was done on 2014 and 2015, and these assessments actually are outdated because since then Nanopore has released new chemistry and new flow cell, and this is one of the reasons we wanted to do that. So what we found is that Nanopore have several errors, so what we call the general error, which are single nucleotide deletions or indels. As you can see here, 3 nucleotide deletions on base mismatches. So this error actually remains. When we compare that to the Illumina, we don't see that at all with the Illumina sequencer. The main drawback of Nanopore is the homopolymer gaps. So for instance, when you have a homopolymer, so here A, because Nanopore does not read nucleotide by nucleotide, but it reads 5 mer by 5 mer, the sequencer, I mean, the algorithms cannot segregate 5 mer to 6 mer to 10 mers, etc., etc. And this is the reason why this homopolymer is wrongly assessed. Sometimes you have a gap of 6, sometimes you have a gap of 21, etc., etc., because this is one of the limitations of this technology. And similarly, it's that is not limited to homopolymer, but also to dinucleotide repeat. And you can see here when you have these GT repeated elements, all these regions, and same here, GAGA, the read actually are poorly assessed. So to conclude, this technology is still not reliable for small nucleotide variant calling, but we think that we can still use this technology for large fragment when you know actually the limitations. You can actually filter for that and find interesting results. So in brief, we have assessed different software, so different versions of the software, and we always compare that to Illumina sequencer. And we do see that at the very base scaling assessment of using clinical samples, so Illumina is still better, so there is no doubt about that, regardless of which technology you use. When we did also some simulations, we found that we were able actually to see what was actually the best software to align. So Minimap, we compare Minimap 2, HS-BLAST-N, and NGMLR. So our software actually, we focus mostly on both Minimap 2 and HS-BLAST-N. And also we did also some QC on the distribution of length. So what was the most optimal length to sequence? So I'm not going into the detail because here everything has been published. So you will be able to see that on the publication. So what was not published is that. So when we assess the different flow cell And we see that since 2017, when we first started this project, Nanopore did incredible effort in increasing the throughput of their sequencer. So with the new flow cells, so the latest one that we use, so now they have actually a new one, the version 10. So that was the version 4.9.4. You can see in terms of number of basis sequences, they from here to here, they multiply almost by 5 the throughput, which enable us to do a proper whole genome sequencing at a low throughput. And this is what we show here. So with the 2017 versions, with one flow cell, we were able to reach only 1.8 depth of coverage of sequencing. So we can sequence less than 2 genomes. Then with the 2018 versions, we were able to sequence more than 4x the entire genomes. And now with the 9.4, with the same cost, we are able to reach more than 11% of the genome. And depending actually the number of pores you have, etc., you can reach now from 10 to 13x of coverage, which is, you will see, enough for a proper structural variant coding. So the cost actually continue decrease. So here, that was actually some cost for what we have done in the past, but roughly, if you want to do this kind of sequencing, it will cost you $1,000 per genome. So what we have done is we have developed a new tool. So this tool is called Nanovar. It's optimized for Nanopore, but works for PacBio. And what this tool is doing is taking 1 to 2 microgram of DNA. And then we use actually this pipeline, Minimap, we do the variant read characterizations, and then we reevaluate that with HS-BLAST-N. And our pipeline is looking for the insertions, as I said, tandem duplications, translocation, deletions, and inversions. And then we use a neural network, so which is artificial intelligence techniques to assess the precision, the reliability of these structural variants. And this is where, I mean, this neural network ends with a score of a confidence score. At the end of the day, the tools output actually a few files and figures for the QC. So one of them is these figures which show the confidence. So each dot represents one structural variant. The confidence score, you have that on the y-axis, and on the x-axis, you see the break-end ratio, which represents the heterogeneity level of the structural variant. For instance, if you are here at 1, that means that all the reads that has been sequencing has the structural variants. And if you are here at 0.5, that means that only 50% of the reads have the structural variant. So and here you can see that we are able to assess the level of heterogeneity of the structural variants across all the cell populations that we are sequencing. So same here, just an overview of how this neural network is working. So basically, what we do, the neural network actually input features are this one, and everything that you have here in parentheses, number of features for the neural network. So to make it short, we take into account the breakpoint alignment features, which include aligned or unaligned proportions surrounding the breakpoint, the alignment e-value surrounding the breakpoint, etc., etc. We take into account also the complex alignment features, which can be the total number of sub-aligned processes by reads, etc., etc., and also the read depth feature. So how many reads confirm this depth of coverage. And all these, so we have all these components in our network classification, and that allows us to improve the variant coding accuracy, use a lower amount of reads, and lower the cost and the labor. This software has been published last year in Genome Biology and is already accessible online. And we, so if you want to use it and to continue to develop that, everything is available in GitHub, also in Python index package. But if you want that as a full package without any installations, you can actually just copy what we have in Bioconda, and everything will be on it. So we have benchmarked Nanovar against other software, and with 4x to 12x, so with homozygous and heterozygous simulated dataset. And as you can see, in terms of recall and precision, we always reach F1 score greater than 0.9. So which means that in terms of recall and precision, we outperform from far all the other software as we are here, and you can see all other software are there. And we also compare that with Illumina-Seq. And when it comes to structural variants, the second generation of sequencer, so Illumina-Seq is here and here, we also outperform that particularly in terms of recall. And that is also true when we are working using heterozygous data sets. So, and we did that with 4x, 8x, and 12x of coverage. And regardless actually how you set up the experiment, NANOVA actually provide much more high accuracy and high quality structural variants than existing tools. And the reason why is that existing tools were optimized for high depth of coverage, and NANOVA actually is optimized for low depth coverage. And this is the reason why we outperform that when we do that at the low depth of coverage. So when we segregate all these structural variants by families, so duplications, deletions, insertions, translocations, and inversions, we can see that for all these categories, when we plot the F1 score, so this is what you have in this spider plot, NANOVA outperform also all the software, and in terms of structural variants, it also outperform what we were able to do with Illumina, with a high depth of coverage. And you can see that Illumina is not able to find big insertions, and that is expected because by definition, the second generation of sequencer rely on our reference genomes. And if the sequence is not in the reference genome, all these insertions won't be able to be brought to light because they are not in the reference genomes. And this is the reason why the F1 score here is close to 0. It's just because this the short read sequencing is not optimized for long insertions. And this is one of the reasons we outperform that. So we do that very well in homozygous and heterozygous in a heterozygous setup. So we have tested that on patients. So 2 AML patients, we have found several structural variants here. So here I just highlighted a few of them. So we just random pick some structural variant and did some molecular biology validation. So, and we choose different scores and different break-in ratio. What you can see first is that our break-in ratio score, so when you go from here to 1, from 1 to 8, really recapitulate the heterogeneity. And you can see that in this plot. When you have only one, when the structural variant is present in all cells, you have only one band. And more the score decrease, more you will see a second band. So the intensity you see of the blot correlate very well with the score that we assess here. So which means that some structural variants actually are homozygous, some of them are heterozygous. We can validate that, and the score that we predict is in concordance with what we were able to validate. So just to give you a figure of the different, how a structural variant can be characterized by the different technologies. So with the nanopore, as you can see here, we have a deletions of 500 base pair deletions. We were able to find actually very well the boundary of these deletions with a very long read. So here you can see we have a read of more than 15 kb. When you have with the second generations of sequencing, you are able actually to sequence very, very small reads, and it's a bit more tricky. So here we have aligned that against it here. So just to show the differences. And then with the single sequencing, which is the first generation of sequencer, the only thing that we can do is validate the breakpoint. So you need to know exactly where the breakpoint is. And this is what we have done here. So we have applied that to a larger cohort of leukemic patients. So as I said, our aim was to look at only normal patients. So we have sequencing a bunch of normal patients, so 11 in total as a first trial. As a positive control, we also sequence some patients with non-structural variants. So for instance, here, a translocations between the chromosome 8 and 21. And then as a negative control, we also sequenced normal donors. So what we learned from there, is the first thing is that there is an inconsistency coverage across sample due to the technology advancements. So when we compare what we have done in 2017 and what we have done now, we can see the depth of coverage actually did increase dramatically. And when we see, we correlated the depth of coverage against the number of variants, you can see that when you are less than the depth of coverage actually is less than 6, the amount of variant is quite low. When you reach a depth of coverage more than 6, so here it's between 7 and 8, the level of structural variant saturate. So this graph shows us that to have a comprehensive landscape of the structural variants, we need to reach at least, let's say, 8. So this is what we recommend, 8x of coverage. And here you can see that from 8x to 12x of coverage, the amount of structural variant does not increase. So you have actually a plateau here that is reached when you have a 8x of coverage. So what we were able to find in this panel of patients is that the great majority of the structural that we found were deletions, so 43%, then insertions, so big insertions, big deletions, following by duplication. So duplication is a subfamily of insertions actually, and then very small amount of inversions, only 1%, and some structural variants, some translocations, 7%. So we were able actually to validate, to find what the clinical cytogenetic laboratory did. So for instance, all the structural variants found by the cytogenic actually were found very precisely with our technology. And we were also able to see the prevalence of the structural variants across all the cohort of individuals that we saw. And as you can see here, just a few of structural variants are shared with everyone. And the great majority of structural variants are the one that we call the major. So major is present in more than 50%, which suggests that this structural variant actually is not pathological, but probably a structural variant present in the ethnical group. And this is the reason why we found that in the normal populations. Same for the blue one. So only actually a few structural variants here are found. So what you see here at singleton, so which are unique to the patients. And we are currently trying to work on bioinformatics tools, which will help us to rank and prioritize the structural variants according to the fact if they have a high potential of pathogenicity or not. For instance, if they are close to a genes that are known to be involved in a disease, etc., etc. So when we go deeper into the annotations, we can see that 40% of the structural variants affect protein codings. Some of them non-coding RNA, and you see 43% actually are in an intergenic region, so an annotative regions. When we zoom into the protein codings, the majority of these structural variants are in the intron, so just a few in the exon. So we can actually look further on this small fraction to see if this actually can lead to a disease or just provide an advantage to this specific group of populations. And then what we show also here is that some of structural variants also occurs on repetitive elements. So, and that occurs in all categories of repetitive elements. And when we look, we compare that to the known and the catalog variants. So the great majority of them, 80%, overlap a variant which has been already described. And what we have shown here in this cohort, 10% of them are variants that are never cataloged before. And I'm going to finish my presentation with one example that we were very interesting. So which is a variant present in an intron in the DCC gene. So you have an amplification here, and this amplification is specifically to AML. We don't see that in the normal. When we go deep in this sequence, what we have shown is that this amplification actually is actually insertions of a sign element. So this is a repetitive element here. and with a very well-known insertion mechanism, so which we call the target site duplication. So you have actually these two sites which are duplicated in a genome of the patients, and that hosts this sign element. So what we think is that with this new technology, we are able also to trace the transposon that jump into the genomes, and then we can also study how what is the implications of this transposon insert into the genome. So what we are, so why we are interested in this gene? So DCC is called DCC because, I mean, it's meant for deleted in colorectal cancer. It encodes for an H1 receptor. This DCC is also associated with normal hematopoiesis. So that actually for us was a smoking gun, because you don't see this in normal patients, but you see that with leukemic patients. So, and this is one of the reasons we are investing in that. And DCC expression is found to be absent in some subtype in AML. So, which we suppose, our hypothesis is that these insertions may affect the expressions of these genes. So in summary, I show you actually two kind of results. So some results derived from a dry lab, so the bioinformatics one, some from my molecular biology labs. And on the drug lab, what we conclude is that despite the higher rate of the nanopore technology, we found actually this technology very adequate when you reach a depth of coverage of, let's say, more than 11x. To study long structural variant. We have developed an accurate low-depth third-generation sequencing long-read variant caller called NanoVar, which is available, and we are working now on a new version.
Sorry to interrupt you. Could you finish?
Yes. So, and then we found that we work that. On AML, and we found that this technology can be very useful for disease that doesn't have many non-genomic aberrations. And this technology is able actually to highlight genomic aberrations that were not detectable by a previous technology. So I just want to acknowledge all my team, particularly the people who were involved in this project. So Tam Chen Young and Jeff Woodlands. And also, so the one that I presented before, also my international collaborator from Paris and Singapore, and also from Canada, and all the funding agency. Thank you for your attention.
Okay, I have just one question, which was transferred to me by Oleg Gusев. So the question is, Toati, I like the tools for low coverage analysis. Do you feel it is ready to go to clinic in cost-effective manner?
So what we are doing right now is we are assessing that, and a few other clinics in Europe are doing that. So what we try to do is to compare what we have with this with a cytogenic. The first result are very encouraging, and it at least can replace the cytogenic assay. And we are actually now, so we have sequencing just a few dozen of patients. We need much more to do that. And we were focusing only on AML. So now we are doing that in other cancer, like colorectal cancer. And all primary data that we have generated say yes. And I am extremely confident also because the chemistry of nanopore is keep improving, and the cost also decrease over the years. So what I can forecast is that in, let's say, 2 years from now, we will be able actually to use that in a clinic very efficiently.
Okay, thank you.
I'm Flora Vaccarino. I'm at the program in Neurodevelopment and Regeneration and the Child Studies Center and Department of Neuroscience at Yale University. And it's a pleasure to be here. And I'll be speaking about modeling human nervous system development using organoids. Okay, shall I share my screen? All right, thanks for being here. So, okay, so there are inherent challenges in understanding the complexity of neuropsychiatric disorders, and most of them are rooted in our own genomes. Can you hear me, by the way?
Yeah.
Okay. There is a large number of personal genetic variants, about 3 to 4 million for each one of us. They could be germline, they could be de novo, and they could also be somatic variants. And these variants impact both the normal and the so-called abnormal brain development and function. And therefore, we believe that brain development based on this genomic variation is likely to be different across individuals. So for example, there may be rare and common variants that may be implicated both in normal development and in disease, and they may contribute to changes in the central nervous system architecture and cell types. However, the relation of these variants to function, brain structure and function is still unclear. And in particular, it makes particularly difficult to link human gene variation to phenotype, clinical phenotype. It could be adaptive or maladaptive. So there are many steps in between. So far, we've tried to link clinical phenotype to human gene variation by doing genomic studies, but that's been imperfect, to say the least. What's been missing is the immediate consequence of human gene variation, that is the effect on RNA and the regulation of RNA transcript by epigenetic phenomena. So obviously, the RNA will be directly linked to building of the CNS, number of cells, number of connections, functional circuitry, which in turn could be more easily linked to clinical phenotypes. So it's been pretty clear that it's central for the understanding of normal and abnormal development to understand gene expression during development. But this has been a particularly tough question because human development is inaccessible and has to be accessed retrospectively. So until very recently, very little has been done in this domain, but two things happened not so long ago. The first one, in 2008, people in the Sazai's lab published the formation of what now it's called organoid, that is the development from stem cells of a self-organized system of aggregates that recapitulate early stages of CNS development. And then, and you can see actually them here, these cells self-aggregate, they make primitive layers, and they differentiate into neurons that are typical of those present in the cerebral cortex of the human brain. And then the second phenomenon was the discovery by Yamanaka et al. In 2007 of the induced pluripotent stem cell technology, which allows the reprogramming of somatic cell from any person into a stem cell, which then could be used to potentially generate such models of human development. And indeed, in 2012, our group used induced pluripotent stem cells for the first time and tried to develop these cells into organoid using the Sazai's protocol. And you can see here, back then, we were able to very amazingly so sort of develop these aggregates from human cells, human-induced pluripotent stem cells. And one important feature is that these aggregates allow morphogen gradients to develop and to maintain cell-to-cell interaction that are typical of human development. So from then on, both us and other investigators have used these cells, these induced pluripotent stem cells, in a variety of ways. But just in general, what you can do, you can access skin cells or other cell type like blood cells from any human donor. These cells will maintain the personal genome of that person with all the variants we've been talking about, reprogram them into stem cells or induce pluripotent stem cells, and then form from these cells these organoids in vitro, which then can be used as models of pluricellular models of human brain development, and can be investigated with a number of assays. For example, you can look at the actual genome sequence, you can look at the transcriptomes, and you can look at epigenetic information using a variety of assays like ChIP-seq and ATAC-seq. And you can also look at DNA architecture and link all this information together to derive gene regulatory event during early development, which is what we were hoping to do. So hopefully, our thinking is that this modeling of early human development may potentially address some of the challenges I was talking about before, and particularly the fact that some of this variation is actually present in non-coding element and may affect gene expression indirectly, but also genome architecture. And there could be multiple relationships between genes and enhancers. So this is not a one-to-one relationship, but you may have many enhancers that regulate one gene, and one gene may be regulated, and one gene may be regulated by multiple enhancers. So the advantage that these induced pluripotent stem cells and organoid can represent is that it allows a longitudinal analysis of the same individual genetic background across development. It can be performed in living individuals, and therefore any finding can be correlated, any biological finding can be correlated with clinical phenotype, and can allow us to understand early stages of development and brain patterning that may be involved in disorders. But one critical question, of course, is whether these organoids are actually true models of human brain development. And in 2018, we addressed this question directly by comparing directly fetal human specimen with organoids. So what we did, we used 3 human fetal specimen, mid-fetal development. We had the cerebral cortex post-mortem from the specimen, and we also had skin cells from the same specimen. So we took the skin cells, we developed them in iPSCs, we developed cortical organoid from these induced pluripotent stem cells, and we cultured them in vitro at 3 and analyzed them at 3 stages of development, TD0, TD11, and TD30 by both RNA-seq and ChIP-seq. And we compared the data obtained in organoid in vitro with the actual cerebral cortex post-mortem obtained from the same specimen. And the data set was relatively large. And one of the first things we discovered was that both looking at transcriptome and enhancers, there was a close correlation, a segregation, I should say, by a hierarchical clustering of transcript and activity of enhancers according to what we were looking at. For example, here you see the organoid on one side and the brain on the other, suggesting that there were fundamental differences between actual brain and organoid grown in vitro. So to understand that better, we compared this data to a large data set of human brain samples. This is a transcriptome data set developed by the CycleCode project, also called BrainSpan. It's over 500 brain samples of all kinds of ages. And we correlated both brain and organoid transcriptome to this large data set. And you can see here on the right that the 2 brain actually closely correlate with human brain sample of their stated developmental time point, which is 16, 17 post-conceptional weeks, whereas organoid correlate best with much earlier stages, and in particular, most strongly at 8 post-conceptional weeks, 9 post-conceptional weeks, and perhaps even earlier. So what that suggests is that these organoids actually represent much earlier stages of development. And this could be important, right? Because these stages of development are virtually inaccessible using post-mortem brains, even fetal brains. So here you see pictures of the organoid themselves. They grow from day 5 to day 30, and past, they get bigger. And you can culture them by the 100. If you section them, you will see them represented early progenitors of the cerebral cortex, which you can see here in the ventricular zone. You have early cortical plate neurons, and you have a variety of so-called radial glial cells that act as progenitors of cortical development. And they actually express, they differentiate into neurons, cortical neurons, and they express the classical marker that we see in cortical neurons of the human brain. Some of them are shown here. They also developed inhibitory cell lineages, which are an important counterpart of the excitatory neurons. So they actually show, the major lineages present in the early human brain. So using this model, we obtained and analyzed a large data set of families with autism spectrum disorder. And within family, we compared the proband with the disorder with their father or with their sibling, which were unaffected. And then we generated data sets of both bulk and single cell RNA sequencing and ChIP-seq, which is a way of looking at histones to map enhancer elements, which I'll tell you in a second. So this is how we use the ChIP-seq data. So here, ChIP-seq is basically an assessment of histone mark. It could be acetylation or methylation of them. And then after you assess this mark on the chromatin, the combination of those marks by comparison with ENCODE data set will actually define a particular element. For example, you see a bivalent promoter as this combination of mark, and a bivalent enhancer or an active enhancer is another combination of histone mark. And so by doing this iteratively, this analysis, we can classify non-coding element in enhancers, repressed, and mixed. And using this method, we defined about 400,000 putative enhancer elements, and we linked them to genes by either proximal interactions, by vicinity, or by taking into account a three-dimensional loop of chromatin from external data set. And by doing that, we derived a secondary data set of about 96,000 gene-associated enhancer elements. And the power of this analysis is that you can actually test, once you have a putative gene linked to that enhancer, you can test what relationship there might be between genes and enhancers by doing correlation analysis. And here on the left, for example, you see what we call an active regulator, that is an enhancer element, which is directly correlated with the RPKM of gene expression, right? So the more active is the enhancers by histone marks assessment, the more overexpressed is that gene. Vice versa, on the right, you see the converse relationship, where the more active the enhancer, the more downregulated is the genes. And so we call these enhancer repressive regulators. And one thing you can do with this data is, because of their complexity, you can't obviously look at one gene and one enhancer by themselves, but what you can do is actually group them by similarity of expression and activity into so-called modules by using a software called WGCNA, which is a type of network analysis. And as an example here, you can see MG19, MG6, these are all gene modules. And in the heat map, this is the actual RPKM or gene expression. And these modules are grouped by similarity, that is, they all share certain patterns of expression over the sample data set that we have. For example, in the neuronal, the modules annotated by neuronal genes, you see they tend to go up during development between TD0 on the left and TD30 on the right, whereas some modules that are associated with cell cycle annotation tend to go down during development, which is expected because cells progressively become post-mitotic. And similar relationship over time you can identify in enhancer. However, while we could see differences across individuals, we couldn't detect major differences in the eigengenes of these modules, gene modules or the enhancer modules between patients and controls. So this analysis in both tissues have certain limitation, which is probably due to the heterogeneity of the organoid themselves. So in order to address that, we developed a single cell data set. That is, we actually analyzed gene expression and enhancer activity within single cells. So here is an example of this analysis on the left. You probably have seen this type of analysis. Each dot is represented by a cell in this so-called UMAP plot, and they are grouped by similarity in their transcriptome. So each so-called group numbered here like 12, 5, 23 represent cells that are similar to each other with regard to their transcripts. And you can also assess the potential bisimilarity, again, a potential time course between this grouping, where you go from progenitor cells in one into various type of intermediate progenitors, and then excitatory neuron and inhibitory neurons. And you can annotate these cell groups by using canonical markers of brain development. And by doing that, you can actually identify the cell group that are present within this cluster. So for example, here RG is meant to be radial glia, these progenitors I was talking about before, and then you have intermediate progenitor, and then you have excitatory and inhibitory neuron, again, defined by particular classical markers of gene expression, which are listed here. You can also compare these cells to the actual human brain, and here you see a correlation analysis between organoid cell group and fetal brain, and we identify similarities between them by correlation analysis between cluster markers, and these are radial glia that correlate with each other, excitatory neuron and inhibitory neuron, suggesting that at least in a broad sense, there are similarities between organoids and fetal human brain at the single cell level. And again, even here, like in the both transcriptome, we can identify lineages, right? And here you see on the top an excitatory neuron lineage represented by 5 such subclusters. You see them here in blue. They express typical markers of excitatory neuron. And in brown, you see inhibitory lineages that are in an adjacent cluster, which express marker for inhibitory cells. Another interesting property of this analysis, characteristic of these cells, is that we can also identify primitive regional specification of the human brain. So here you see a gene called FOXG1, which is an early determinant of cortical development. It's very strongly expressed only in the telencephalon, but not in primitive regions of the human or mammalian brain. And you can see that it's actually expressed in most of these cells, except for 2 groups here on the right and on the left. And these 2 groups that don't express this gene actually express a series of markers of so-called cortical hem and medial cortical plate that are 2 separate areas that are adjacent to the cortex, but not quite as similar to the cortex. They're more primitive cells, and they're characterized by a set of genes called Wnt genes. And in mouse, this has been investigated many years ago, where they found that these Wnt genes indeed are not in the cortex, but they are in a region adjacent to it. And so suggesting that Wnt inhibition is required to produce the dorsal cortex. Okay, this I'll come to that in a few minutes. So one thing I wanted to point out is that these various cell types are there in the organelle, but they're quite variable, and you can see them here. This is the proportion of different cell type in various color, with the excitatory neuron, the radial glia, etc. And in each line, you see a particular sample, and you can see that this representation is not the same, even though you're looking at stages, very late stage here, TD30 and TD60, you have quite a large variation. You can see here, for example, that the inhibitory neuron and this medial cortical plate I was alluding to before, characterized by Wnt signaling, is also quite variable across samples. So it's not easy to actually assess number of different cell groups or cell type across individuals because of this variation. However, you can still look at gene expression within each particular cell type. So again, here there is no specific characteristical cell proportion that distinguish the autism cell in the autistic individual from the control. And so we resorted on analyzing gene expression within each cluster. And if you do that, you do see some tendencies, differential gene expression across patients and control. For example, here within the excitatory neuron, patients, this is a volcano plot, and on the right, you see the overexpressed gene. You see that patients have an overexpression of certain genes in the excitatory lineage and a decrease in the expression of Wnt gene, which are in the medial cortical plate. And the same is happening in excitatory neuron progenitor. So it's feasible using this model to actually look at differential gene expression, if not differential number of cell types. However, in the last few minutes of my presentation, I wanted to go over another model that we've been developing. And this is something that we've been thinking about for quite a while, and that arises from the fact that these organoids are actually not the same. You can develop, many groups have developed organoids of different types. So for example, you can use a default approach here on the left, where you basically leave these cells develop as they do spontaneously. And if you do that, they'll develop a variety of different cell types, from retina to hippocampus to forebrain to choroid plexus. And so you have quite a large variation within each organoid, and that precludes some of the assessment of gene expression that one can possibly do using this model. On the right side, I'm showing you two examples of what I call time cookbook approach. That is, you actually give to this organoid in vitro certain morphogens, certain compounds that will directly push them in a certain direction. So on top, you see hippocampus, hippocampal neurons, and on the bottom, you see the optic cap, the eye development. So, and this is dependent on what you actually add in vitro to the culture media. However, this is not what the brain, how the brain develops, right? Because normally the brain, this is an image of early human mammalian brain, develop, you know, you don't, there is like a graded developmental phenomenon here, where you have morphogen that spontaneously diffuse in the early epithelium. You see on the roof, here on the left, what's called roof plate, which is a source of Wnt, which I've talked about before. And on the bottom, another molecule called sonic hedgehog, which is at the basal region of the CNS, and they form gradients. And these gradients mix with each other over development and generate the various regions of the CNS. So it's a graded approach. It's not an all or none approach. So we thought whether perhaps this could be a better way to model human CNS development using this graded approach. So first of all, I wanted to again reiterate that Wnt inhibition is required for producing the cerebral cortex. And that's what we rely upon in all the organoids I was showing you before. We add a Wnt inhibitor to begin with to allow cortex to develop and inhibit the medial cortical plate, right? And that's what I've been talking until now. So what we thought that instead of inhibiting Wnt, we add a Wnt gradient to the system using a spatial chamber, which we developed in collaboration with the Yale Bioengineering Department. And so by adding so there are two reservoirs here, and we add a Wnt agonist on the right reservoir, and that will make a gradient across these 5 chambers. And each chamber will be exposed to a specific concentration of this Wnt agonist. And the idea was that this Wnt agonist would actually pattern the brain in an anterior-posterior domain, because the anterior end must avoid Wnt, whereas as you increase Wnt concentration, you are progressively specifying posterior regions. And how are we going to read this gradient? Well, one way obviously is to use gene expression, and we use the variety of genes that have differential expression in the anterior posterior domain. For example, you see OTX2 in yellow, that's expressed until this region here between mesencephalon and rhombencephalon. And then you have another gene called PAX2, right, which is expressed in the posterior region up to the mesencephalon and the diencephalon. If in the diencephalon anterior to that, you have PAX6. And then you have 6-3, which is the most anterior one, and FOXG1, which is the one that is anterior most of all. So using this gene expression by qPCR, we compare the bath application of Wnt agonists to the actual chamber exposure to a Wnt agonist. And in fact, what you can see here is that we were able to reproduce, using the device, a graded topographical representation of anterior posterior regions of the brain. And you see forebrain here with FOXG1, and then midbrain, which is characterized by OTX2 up to here, and then hindbrain, which has neither of those, but has things like Wnt1, GBX2, and IRX3, right? And this is true no matter what we tested with using different media. This is hydrogel, and the right is collagen. Depending on the media, you can shift the boundaries, but you pretty much have the same patterning occur using this chamber. And here is the Wnt agonist I was telling you before. And this was validated by immunocytochemistry, where we saw similar expression of the protein themselves in the corresponding organoid. So with this topography, we could actually reproduce a very important boundary in the brain, which is called the isthmus. And you see this here in a mouse brain and below in a human brain, where you have OTX2 expressed up until the isthmus and GBX2, which expressed down in the spinal cord and the brainstem up to the isthmus itself. So this is basically the major division between brain and brainstem and spinal cord. So it's a fundamental boundary in mammalian brain development. You can see the boundary reproduced here using the gradient in our chamber, where you have that between C2 and C1 chamber, OTX2 that is up, that's the anterior gene, goes down in C1, and correspondingly, you see that the GBX2, which is expressed posteriorly, now goes up at the same boundary where OTX2 goes down. And again, the boundary shift depending on the media, the collagen or the hydrogel, but is still reproduced quite faithfully using the chamber. So it's characterized by these two fundamental genes. And together with GBX2, which you see here in green, you have also expression of a variety of Hox genes, which are homeobox genes expressed in the spinal cord and brainstem. So one of the most interesting things is that these boundaries are reproducible across people. So here you see not just one IPS line, but several of them, each them represented by a different color. You see them up here with GBX2. So they're slightly different, right? So that goes back to my initial point that development is not identical, even in this very early fundamental step across people. And you can see here that each person still follows the same trajectory, but in perhaps slightly different way. Okay, so you have GBX2 going down from posterior to anterior, and OTX2 going up from posterior to anterior, and FSF2, which is a cortical marker, also going up from posterior to anterior in slightly different ways. And finally, I want to close by saying, I'm not going to show you much data for this, that we're now going even more trying to advance this concept by developing a dual gradient, because we don't only have anterior posterior, but we also have dorsoventral, which is quite important in patterning the early brain development. So we are developing a dual chamber where we have not only a right-left sink and reservoir for Wnt, but we also have a parallel reservoir in an orthogonal direction for sonic edgio, which is an important morphogen that specifies the ventral brain. And here you see the simulation, a computer simulation of the diffusion of these two gradients. One is here and one is here in the two orthogonal direction. And we have preliminary data suggesting that indeed we can achieve not only the anterior-posterior specification I was alluding to before. For example, you see 6.3 here, which is an anterior gene, and is present in both the dorsal and ventral chamber, but you also have dorsoventral specification. For example, here you see NKX 2.1 that is only expressed in the posterior domain, in the ventral domain, but not in the dorsal domain. So so that's basically comes to my conclusion. Organoids reproduce early telencephalic architecture, precursors, which are called radial glial cells, excitatory and inhibitory lineages, and regional specification into neocortex and archicortex, and can be used to understand gene expression and regulation in both normal and disease states, right? However, there is a variability in differentiation and in cell type number across individuals, possibly driven by genetic background and maybe other stochastic factors. And however, topographical patterning, which mimic the embryonic axis development in the real brain, may reveal a different response in regionalization across individuals and perhaps can provide us a more consistent model of human brain development. And finally, I want to acknowledge people in my lab that have been working on this modeling over the years. We've been working on this for over 10 years now. In particular, I'd like to acknowledge Alex Jourdan and Jessica Mariani, Feinan Wu and Scott Norton, which are two brilliant bioinformaticians. And of course, Alexei Abizov's lab, who has collaborated with us over the year in a number of projects. And the Levchenko lab, who has helped develop these three-dimensional chambers I was talking about. Our collaborators at the Child Study Center that helped us recruit patients to develop iPSC's line, and finally our funding agencies, including the NIH, the Simons Foundation, and the Kavli Institute for Neuroscience. And with that, I'm going to stop, and I welcome any questions that people may want to ask.
Sorry, I have just one question.
Sure.
So, is it possible, do you think, to predict the phenotype of an organism using the genotype information only? Is it possible in principle?
Well, this is a very difficult question, because if I answer yes, that means that I believe that the genotype is the only determinant of the phenotype, which is the clear answer to that is no, right? I mean, there is a genotype that has a profound influence on the phenotype, but there are other things which derive from the interaction of cells, the microenvironment of cells, the communication between cells that have a certain stochasticity, and it may be guided by environmental factors as well, like in the macro environment, say in the intrauterine environment, for example. And so it goes back to my statement that the number of cells of particular cell groups is variable across individuals, and that's not dependent, we know that is not only a factor of genetic background, there are other factors.
Okay.
And so I suspect that that's true also for human development in vivo. Yeah, it's quite interesting that we can at least prefigure some aspect of human brain development based on the genotype of the cells. Any other question?
Nikolai?
Yeah.
One again for the invitation. Shall I leave the room?
Okay, you can leave the room.
Thank you. Bye.
Bye.
Окей. Я не знаю, на каком языке лучше говорить, потому что я не знаю, какая аудитория. Лучше по-русски или по-английски?
Смешанная аудитория.
Смешанная.
The audience is mixed, so English is preferable.
Okay, well, we'll do English. Потому что, честно говоря, тяжело по-русски, потому что терминов нету. Если вы посидеть с Дмитрием Быковым пару дней, повыдумывать новые термины, это вряд ли произойдет. Okay, so I guess I'll start in English. Let me share the screen. Okay, so do you see my slides?
So the last speaker is Dr. Anton Nikrutenko, the global system of data analysis in a public scientific cloud.
Thank you. Thank you. Thank you very much for inviting me. So this is sort of a Russian translation of what I'm going to be talking about. I think I invented the term public node, публичное научное облако. Well, it's okay. So it's a global system for data analysis, for cloud-based system for data analysis, and SARS-CoV-2 as the engine that drives the development. So throughout this presentation, there will be these URLs. You can go to them and sort of see what I'm talking about. If it's possible, I sent the link to this presentation to Yulia, and if she can share it with them, so it can be shared if anybody's interested. Okay, so I come from Kiev some time ago, and then I moved across here. Well, actually, right now I'm closer to Moscow than US, but anyway. And right now I'm at Penn State. So at Penn State, we run a project which is called Galaxy. It's kind of there is a history to that name. And so I'll just briefly explain what this is. So this is it's many things. It's a software framework which is designed to allow people to analyze very large collection of samples via UI. You can do it through API as well, of course. But it's really the only place where you can go and analyze hundreds of thousands of samples through user interface. It's a very hard problem, and in general. And so we have, I think, a good solution for this. And because modern sequencing is cheap, very few people sequence just a few samples. Most people sequence hundreds, perhaps thousands of samples. And in the title of this talk, I was trying to say that SARS-CoV-2 is sort of an engine of progress. And it's because it's the first pandemic which coincides with relatively cheap sequencing. So it's the first time that we see data sets of this size. So it's a good test for many software tools. For example, how do you exactly build phylogenetic trees when you have 1.5 million sequences? How do you do selection analysis when you have many sequences like that? How do you reliably monitor emergence of new variants where you have continuous stream of sequencing data sets and so on? And it's actually a lot of software which we considered good before doesn't really scale. So it's an interesting problem. And so in that sense, I guess SARS is a good thing, scary thing to say. So it's also a workflow system. So essentially it's an execution system which allows you to run tools on data. So we have a workflow language. This is, for example, this is an example of one of the SARS workflows that I'm going to be talking about. And so it's also engineered to run on latest modern high-performance compute infrastructure, and that includes public and private clouds. So we have some instances which run on Amazon, some projects where we use Google. However, really the subject of this talk is about free system which uses public clouds. And this is very important because when you talk about compute clouds, you usually think about Amazon or Google or Microsoft Azure or other smaller systems. But the problem with these clouds is that they're wonderful for doing, for example, many types of data analysis problems that we have in biology, but they're not free. And if you live in the US, for example, you work at the US university or at a research institution, you perhaps can pay for that. But if you're, I don't know, in Russia or Ukraine or somewhere else, even in France, for example, it's difficult for many reasons. Some of the reasons are purely protectionists. These are American things. So for example, Europeans are much less likely to use them. And some things are just, paperwork stupidity of this sort of that, how do you reconcile the bills? So why we have a lot of systems which rely on commercial clouds, it's not a good solution if we're looking to give the entire world an ability to analyze large data sets. And so there are several types of free public clouds that exist. So in US, there is a system called Exceed. It's essentially a network of multiple supercomputing centers, such as TAC, which stands for Texas Advanced Computing Center. There's also Jetstream Cloud. These are all projects funded by National Science Foundation. And so I mentioned that Galaxy is software. So this software can be used to run Galaxy instances. These are actual websites where you go and analyze your data. There are many instances, but there are three big ones which operate in, this is a large collaborative effort. So one in the US, one in Europe, and one in Australia. And they all rely on public clouds. In US, that's Exceed that I mentioned. In Europe, it's part of the German network for bioinformatics infrastructure, also Elixir, which unites a number of European countries and also provides infrastructure. And in Australia, it's Australia-specific Nectar Research Cloud. So these are all examples of free public clouds that researchers can use, and they can use them across the boundaries. So, if there are kind of doctors without borders, these are clouds without borders in some sense. There's also a number of local instances, for example, through Europe. We don't really track that. So Galaxy doesn't call home. So there are hundreds of instances, but these are the ones that we're actively working with as well. What I mentioned so far, it's a software. You can install that software. It would allow people to run open source, well, actually any tools. We, of course, in public instances, we only use open source tools, but there are proprietary Galaxy instances which use proprietary tools. And so you cannot do any analysis without tools. And Galaxy embraces, have tight relationship with Bioconda. It's probably the biggest effort to simplify installation of open source bioinformatics tools. It's essentially a dependency management system, but it's a version dependency management system. So if you, if you run some old version of, I don't know, BWA and you want to rerun it 5 years from now, you can do that because all the versions are preserved here. We're also collaborating with Biocontainers, which is an effort to containerize all these tools. And so again, to kind of standardize their execution. This is all about making data analysis reproducible, predictable as well. So this screenshot here, this is our app store. It's called Galaxy Toolshed, and this is where all global Galaxy instances pool their tools. But that system relies on Bioconda for dependency resolution. We also have comprehensive training network. It's a community project. If you remember the first slide, it had this cloud of different organizations. It really spans US, Europe, Australia, Africa. So there are a very large number of contributors. And so we developed these training materials, and they range from sort of basic, how do I analyze the data, to more of the more complicated things, such as, for example, examples of particular types of machine learning analysis and so on. And we run a number of webinars. This slide is again focused on SARS-CoV-2 because that's the game of the day. We even published a few papers about how to teach during the lockdown because what else to do? So motivation behind our SARS effort is this. If you look at the landscape of coronavirus data analysis, it's not better than when you read the news from different places. It's all very confusing and it's hard to know what is from where. So at the research level, at our level, it's unfortunately the same. So SARS-CoV-2 data analysis is very fragmented. You have large institutions that know what they're doing, and so they're performing reasonable types of analysis. These institutions, probably the best effort is the one in the UK, called UK effort. That's the group of people that know what they're doing. But it's a power law distribution. So you have these large groups, then you have all sorts of smaller groups in different countries, and then it becomes a little bit more complicated. And it's not to blame anybody, it's just imagine yourself in the shoes of somebody in, I don't know, in Tunisia, trying to process samples from a local hospital. What do you exactly do? What kind of system are you going to use? Well, again, I just mentioned the problem with the clouds. So if this hospital generates a lot of data, you don't really have infrastructure. But even if you have infrastructure, how do you call variants, for example? And those of you who are listening, well, who are still listening, it's 6 p.m. Calling variants is not a particularly complex problem, but in the past 10 years, most of the effort was because of the efforts such as 1,000 Genomes and other large human pan-genome, all these efforts. We know how to call variants in humans very well in deployed systems, but the simpler actually haploid mixtures such as SARS-CoV-2 were kind of less familiar to the wider cloud. So as a result, we have plenty of papers that use, for example, GATK for SARS-CoV-2 variant calling, which is complete nonsense. But I mean, GATK will call variants. So you will get obviously lineage-specific variants from GATK. But if you're looking at interhost variation, if you're looking for cases, do you have co-infections, for example, then that's not a good approach. And most of the current analysis are focused on generating consensus. And so you're a hospital, you sequence the patient, you generate consensus, you compare against GZ, and you say, okay, well, this is some kind of a derivative of the Delta variant. But that sort of a fine grain, what's happening within individuals, that's ignored. And as I mentioned, the access to infrastructure varies from country to country. So our goal here is to develop a system which can be used by anyone. And so what we've done, we've developed a series of robust workflows. These are validated workflows. They're developed as using proper software development practices. They have tests. They're deposited, they're versioned, they're deposited into proper repositories, and we'll talk about this later. So most of the COVID data comes in the form of amplicon-derived sequencing using RTIC protocols or other protocols. And it comes primarily from two technologies, from Illumina and from Oxford Nanopore. There's some small number of data coming from other technologies, such as Ion Torrent or PegBio, but mostly it's Illumina and ONT. And so as a result, we developed workflows for ONT for RTIC data and for Illumina for RTIC data, and also for metatranscriptomic data. So this is the rare types of studies, there aren't that many of them, when actual samples from a patient are taken and RNA is isolated, and so it's done using RNA sequence, so there is no amplification step, and that's ideal data for looking at intra-host variation. So 4 workflows: Oxford Nanopore for RTIC, Illumina for RTIC, and 2 kinds of Illumina workflows for metatranscriptomic data. And then we also have a reporting workflow which generates outputs which can be used for interpreting these results. And we use, so this is designed to be run on hundreds and thousands of samples, and we operate it in 2 ways. So on one hand, we have public, so anybody who would go to this covid19.galaxyproject.org can run these workflows now. If you have your own data, go ahead and run them. We also run them in an automated way for all the COG UK data, which comes in also for data coming from several countries, which use this as their primary analysis framework. And then we generate dashboards. And for those of you who are not familiar with the notebook environments, probably most people familiar with Jupyter or RStudio, but there are these wonderful new things that are popping up. And one of them is Observable, which is a JavaScript environment, but it's an amazing reactive way to represent results of your analysis. And I'll show an example of that. So workflows that we develop, they're deposited into, there are 2 kind of a global hubs for the workflows. On the US side, it's DocStore, run out of UCSC, University of California, Santa Cruz. It's part of the Anvil project and many large-scale projects on the US side. And on the European side, there's Elixir effort called Workflow Hub. So this is how we used to, this is the outlets we use to distribute our workflows. And well, in the end, this is just, this is data like this. So those of you who used to look at PCF files, we don't, we provide various kinds of outputs, but we keep this all. So we keep the primary data, we keep all the intermediate calls, we keep all the consensus sequences, and it turned out to be extremely useful because if you rely on, for example, SRA or usual places where sequencing data gets developed, sometimes things get deleted, but we actually keep everything from original read level data to all the intermediate calls to consensus sequences, and this is again for hundreds of thousands of samples. And we distribute this data into 3 primary ways. We are partnering with Viral Beacon in Barcelona, which is, we also use the FTP site at Viral Beacon to distribute all the raw data, which I just talked to you about, the read level data and all the variant calls. So if we want to go back and examine this, that can be done. And we also provide these interactive notebooks. And so they look like this, for example. This is an example for Irish data. So Ireland is using us for processing of some of their data sets. So once analysis are completed on the Galaxy side, they can then ingest it into this observable environment. This is this new JavaScript environment I just mentioned. And it's really, you can build amazing widgets here. So for example, let's recompute for a particular slice of dates. You can do, and you can add these widgets as you go. So we have several types of widgets here, just the visual representation of data, variance per gene, the lineage distribution, and also there is an ability to look up individual variants, for example, how the frequency of individual variants tracks across time. So you can just select variant here and do that. So well, that's not particularly interesting one, but there's a number of others. So, well, you get the idea. Also, we keep track of these variants of concerns, the new lineages which continuously occur. And this, for example, this is all the Delta variant sites. And again, you can track, you can see where they're beginning to appear in the data and various kinds of reports. And again, it's open. You can clone, you can fork this. You can add widgets here and so on. So as I mentioned, coronavirus was a wonderful thing in terms of scaling up software. So as I mentioned, we run primary free, plus we have many of the regional instances in Europe primarily. So France, Belgium, Estonia, Spain, Italy, and many others. And so they're doing their individual things, but they all have access to the same infrastructure. They all have access to the same tools. This is done through so-called Pulsar network. Pulsar, it's a system for running jobs, running compute jobs on remote resources, which is developed as a part of Galaxy. It's a system which was also developed by us. So you can imagine that the entire world of computational resources, and these resources only need to run a Pulsar to appear as a Pulsar node, and then any Galaxy instance in the world, whether it's in the US, in Estonia, in Poland, in Greece, in Crete, can schedule jobs on that node. So for large efforts, for national efforts, for COG UK efforts, of course, this is all done automatically. So we have an automated way to trigger these workflows, which I showed you in graphical interface. So we have a way to trigger these things programmatically. And then we keep all the data. This is an example of JSON file with batch IDs, history links, individual accession numbers for COG UK data. This is all stored in Barcelona. So on one end, we're providing dashboards, so something for the general public to consume, as well as we provide all the other information for reanalysis, re-examining things, and so on. And you can do a lot of interesting science in this, unfortunately, which I will not have time to talk about. So that's sort of a summary, is that we take data from national resources. For example, COG UK data comes from European Nucleotide Archive. In US, data comes from SRA. Then we use our workflows, which are in the workflow hub and in the doc store. This is where national Galaxy instances get the workflows. Again, they're versioned. And the outcomes of running these workflows are distributed via, so everything can be obtained as raw data or can be consumed as ready visualizations. So you can, again, if there's a way to send the URL for this presentation to all the listeners of that talk, so these things here are the live links. So you don't have to trust me, because in half an hour, it's very hard to describe complex software system. Normally, I would do a hands-on demo, but it's hard to fit this in half an hour. So maybe what I'm telling you is complete nonsense. Maybe I made all this up. So you can go ahead and try this. For example, this is an example of hands-on tutorials which we develop. And this document describes everything. So you go to, it explains to you that you need to go to a Galaxy instance like this. You can do it in the US, in Europe, in wherever you are. It provides sample data. So this is the set of Arctic data sets. So you can play with this data. You don't have to use this data. You can substitute this with the data sets you actually want to analyze. And then it goes through all the steps explaining. So it's a step-by-step description of this analysis with explanation of all the intermediate data sets. There are a lot of things which I didn't have time to mention, which these workflows generate. For example, variant frequency plots, which are good for looking at limited number of samples. There is also this page, which summarizes our effort and explains how our resources can be used. This is the link to the current dashboard, such as the one that I showed you. So it's the same thing as this. The one thing to remember about observable notebooks is that because they're JavaScript driven, they're actually executed in your browser. So this is a kind of interesting serverless way, because if you were doing this in a standard notebook environment such as RStudio or Jupyter, you need to have a server and a client system, but here everything happens in the browser. And this is the link to the viral beacon, which is, let it load. Which stores all the data from all the samples that we process. And there will be publication coming out in October describing this. Okay, so at this point, let's see, questions?
Okay. One question. Do you think that the evolution of resources will go to the aim of more and more open source resources or more and more payable resources?
By resources, you mean clouds or tools?
Yeah, both clouds and tools and their content also.
Well, the clouds, it's a war, obviously. I mean, there are. I mean, it's hard to predict this, because obviously public clouds that I mentioned, they rely on scientific funding. So they're usually funded by scientific funding agencies in respective countries. But again, that's another kind of a weird effect of COVID. It was good for that funding. So for now, these resources aren't going anywhere. In terms of tools, open source is really the only way, because otherwise, how do you know what it does? So, I mean, if you have a variant caller and it does something, well, maybe, maybe you don't care. I don't know, but I don't think it's possible to evaluate results based on black box magic. So, but it depends on a particular field. In genome analysis in particular, almost everything is open source. It's a kind of accepted practice in the field. In other fields, it's not so straightforward. For example, in proteomics and mass spectrometry, in image processing, that's not as clear. And if we're talking about the future, probably image analysis will be kind of the next big data wave, because sequencing is it's kind of a stabilizing. There will be a lot of sequences, but with these new techniques, as different kinds of microscopy, the cryo-EM, all these things, there will be just enormous explosion in the need for image processing. But there is also a lot of open source development happening here. Of course, I'm very opinionated here. We don't run anything which isn't open source, and we don't need to, because once new technology appears, there will be a number of new tools. Of course, you also need to know, which tools are good, which are bad, but that kind of it can be easily judged from GitHub repositories for these tools. So open source really allows you to gauge things. It's like, в Булгакове осетрина второй свежести. You can tell первой свежести осетрина или второй based on GitHub traffic.
Okay.
So, Nikolai, can I ask? So, Anton, thank you for accepting the invitation. So, I first heard about Galaxy like 10 years ago, maybe even more, being still a postgraduate student, and it was still already impressive. And now, 10 years after that, it's more impressive. But what is your foreseeing for like next 5 years? What will you be investing in the frame of Galaxy, or maybe it will be new products? So, where this development is going? So what is kind of far and near goals for that?
К победе коммунизма. It's hard. Unfortunately, it's hard to think in 5-year terms. But it's hard to sell free stuff. So it's a sort of a constant battle in terms of, for example, these workflows that we are describing here. I mean, they're used a lot, but it's sort of like with vaccination, right? There's some reluctance on using good open source stuff. So there's some people think that if you buy something from CLC Bio, that will be better. It's okay. I mean, but it's kind of the same thing as with vaccination reluctance. It's very similar. So I guess my plan for the next the plan for the next 5 years is to, there's a roadmap. Of course, we need to make interface more robust for really large scale. We need to, and we're working on different ways of representing complex samples, and make completely detach sort of the instance from compute, so it can compute anywhere. Use GPUs. There are a lot of things that we need to do.
Thank you, Adam. Thank you very much.
Thank you. Thank you for inviting me. It's late, so.